Okay.
Okay,
So I
so
think
I
I am
think I am audible now
clearly.
clearly.
Good
Good
evening,
evening,
everyone.
everyone.
So
So
today,
today, since my microphone is not
I
working, I
am
am
using the
using the
audio of
audio
my
of my
mobile.
mobile.
So if
So
you
if you
face
face
any
any
problem,
problem,
then
then
you
you
can
can just
stop me
stop me in between,
maybe,
maybe,
and
and
just
just
ask
ask
me
me
whether
whether
you are
you are
not
not able to, or it is not
clear.
clear, the voice is not clear. Then
Just
just
stop me
stop me in
and
between and tell me that. Okay,
that's
since
why
my there is some problem
with
with I think with
uh, with the
the
microphone.
microphone,
So
so
that's
that's
why
why I'm using my mobile phone. Okay, so
if
if
there
there is any problem, just
stop me.
stop me in between.
Okay.
Okay,
So
so
without
without
wasting
wasting
much
much
time,
time,
so
so let us
start
start
today's
today's
discussion
discussion
session.
session.
So
So
okay, my
okay, my
slides are
slides are
visible.
visible.
Hopefully,
Hopefully,
I can
I
see
can
it
see
on
it
my
on my
screen.
screen.
So
So
yes,
yes, so
good
good
evening,
evening,
everyone.
everyone.
Welcome to
Welcome to
the
the
week
week
11
11
discussion
discussion
of the
of the
structural
structural
biology
biology
course
course
BT28
BT28
offered
offered
by
by
NPTEL.
NPTEL.
So
So
always
always
before
before
starting
starting
the
the
discussion, I
discussion, I
always
always
ask
ask
you to
you to
ask
ask
any
any
questions
questions
you
if you
have
have
related
related
to
to
the
the
previous
previous
discussions,
discussions,
or
or
we
we
will
will also
discuss the
discuss
week
the week
11
11 week 11 assignments
in the
in
end
the
of
end of
today's
today's
2 hour
2 hour
discussion
discussion
class.
class.
So
So
before
before
that,
that,
if you
if you
have
have
any
any
doubts,
doubts,
anything that
anything
you
that you
want to
want to
clarify,
clarify,
anything you
anything
want
you
to
want to
ask, you
ask,
can
you can
now
now
is
is
the
the
time to
time
ask,
to ask,
or you
or
can
you
ask
can ask
me
me in
between
between
the
the 2 hour
session.
session.
So
So
yes,
yes,
anything
anything
you
you
want to
want to
discuss,
discuss,
or any
or any
doubts you
doubts you
have.
have.
Okay.
Okay.
So if
So
there is
if
no
there is
doubt,
no doubts,
then
then
one question
one
I think
question I
that
think
was
that
asked
was
by
asked by Paromita. She
her,
was here, but now
she has
she
left.
has left. So I think she will rejoin.
I
But
just
I
wanted
just
to
wanted to
I
announce.
can
I
repeat
can
it
repeat
later
it later
that
that
she asked
she
last
asked
class
last
about
class
the
about the
exam
exam
pattern
pattern of
that
exam that you are going to appear on 23rd, if I am not wrong,
23rd.
23rd April.
So
So
means
means
I have
I
not
had not
appeared in
appeared
any
in any
NPTEL
NPTEL
exam
exam
before, and
before,
I
and
did
I
not
will
set
not
the
set the
question.
question.
So
So
but
but
I
I
have
have
asked
asked one of my friend who
who
had
appeared
appeared
previously
previously in
in
an
an
NPTEL
NPTEL exam.
exam.
So
So
I
it
think
I think
this
this
varies
varies
from
from
instructor to
instructor to
instructor
instructor
how he
how
or
he or
she
she
wants
wants to
ask the
ask the
questions.
questions.
So
So
in
in
his
his
course, it
course,
was
it was
asked
asked
like
like that
there were
there were
some
some
MCQ
MCQ
questions,
questions,
some
some
multiple
multiple
answers you
answers.
had,
You had some
MSQs were
MSQs
there,
were there,
some
some
MCQs,
MCQs.
most of
Most
them
of
were
them was
MCQs,
MCQs.
some
Some
MSQs were
MSQs were
there,
there,
and
and
where
where
it
it
will
will
be
be
mentioned
mentioned
that
that
multiple
multiple
answers are
answers are
correct,
correct,
and
and
then
and then
you
you
need
need
to
to
answer
answer
the
the
multiple
multiple
questions
questions
in
in
that
that
way,
way.
and
And
also
also
there will
there will
be
be
some fill
some
in
fill
the
in the
blanks
blanks
and
and numerical aptitude based
questions that you can expect that was there in his course, but I'm not sure about your
were
exam
in his
how
course, but I'm not sure about your
designed.
exam how the question paper will be
But
designed.
yes,
But yes,
I think
I think
I
that
can
that
tell
much
you.
I can tell
I have
you.
also
I have
asked
also
one of
asked one of my
friend
friend
who
who
has
has
appeared
appeared for this
for
exam
this
last
year.
year.
So
So
when
when
he
he
will
will
reply me
reply me,
or
or he will
get
get back
me
to me
about
about
the
the
question
question
paper
paper
pattern,
pattern,
hopefully
hopefully
by
by
next week
next
I
week
will
I will
be
be
able to
able to
tell
tell
you
you
about
about
the
the
pattern of
pattern
the
of the
questions that
question that
came
came
last
last
year
year
in the
in the
structural
structural
biology
biology
course.
course.
Okay,
Okay,
the
the
pattern,
pattern,
how
how
many
many means how many
questions were
questions were
there,
there,
and
and
what
what
was
was
the
the
pattern.
pattern.
So
So
that
that
I
I
think I
think I
will
will
be
be
able to
able to
share with
share
you
with you in
uh,
the next the week 12 discussion that we will have. That will be our last discussion session. But again, since I wanted to take a revision class of what we have discussed till now, so on 22nd, on 21st, that will be our 12-week discussion class, and on 22nd, I will take a 1 hour revision class where you can come up with your doubts, as well as the I will revise all the topics that I had discussed till now, so that you can join. And on 23rd you have the exam, so I think that will also help you in preparation of your exam. So these things I
Sir,
wanted to. Yeah. Yes, Sharon. No.
sir, our exam will be conducted on 23rd or 30th April.
I got a mail that your exam is on 23rd, but do you have any notification that it will be on 30? Means I'm not sure. I just got a mail that on 23rd the exams will start, but the exact date of your exam, that I also don't know. I think they will notify you by means by email. I am telling you because on 22nd only I can take the class, because from 23rd the exams will start. Okay, so for that purpose, I thought that
See,
yeah,
till now I could not get my exam admit card.
you could not get your exam admit card. Is it circulated yet? I am not sure about that. Is it circulated? Whether others can confirm whether others had got their admit card or not. I think they have not started giving the admit cards, or because I don't have access to those things, because I will not set the question paper, as well as that is totally on the NPTEL team. So so whether others have got the question papers, or not, others have got their admit card, or uh, can anyone confirm?
No, I didn't get. I think it is
Okay,
not
so
circulated yet.
yes, yes, it's not circulated yet. I think it will be when it will be circulated. I think they continuously email you about or update you about the informations whenever there is some important information there. So when there will be they will be starting to circulate the admit card. I think they will notify you through email, so you will get to know that. And also they will give you the correct exam date through email. But as far as I know, from 23rd, the examinations of means all the NPTEL examinations will start. So the exact date of your examination, that I am also not aware of. I think means I will have to check once, or I think they will notify you. And since
Uh,
yes, yes,
the date has been announced.
it's
It is
23rd,
30th.
right?
It is
It's
30th.
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 It is
30th.
30th.
Yes.
Okay, okay,
Yeah.
okay. So pardon me then. It's 30th then, Sharon. No.
Okay, okay. So,
Okay.
excuse me, Paramita Chaudhuri, are you sure about it?
Yes, a mail has been sent to me. If you want, I can forward you.
Yeah, sure. I'm writing my mail ID in my chat box. Just a
Okay.
minute.
Okay, so I think now the doubt is cleared that your exam will be on 30th. So yes, the about the exam pattern, Paramita, I had asked my one of my friend. He had appeared in one of the exam of not the structural biology course, but in another course. So he mentioned that the question paper patterns that vary to instructor, but mostly they were MCQ questions. Some of them were also MSQs, where it will be mentioned to you that yes, you have to give multiple questions, multiple answers are correct to this question. So yeah, that will be provided in the question only. And some of them were also NATs. So where they will provide you with the information that there is this is a numerical aptitude test, request and how much range you are allowed to give, that they will actually provide you in the question paper. Okay, so that I got to know from one of my friend, but also I have asked someone who had given the previous structural biology course exam. So that I think when he will reply me, I will let you know in the last week. That will be sure. Okay, I will let you know. Okay.
Actually, the mail in the mail where the date is mentioned, they have also mentioned the what kind of question will be given. For particular
Okay.
structural biology course, the exam will be totally MCQ type.
Okay. So then it's fine. So that mail didn't came to me, so that's why
Already
I
we just
didn't.
so we have to just click in the option, right, from CBT mode.
Yeah, I think that will be the pattern. Yes, you have to click on the
Actually,
options.
as far as I remember, there is some virtual calculator will be allowed.
Yes, yes. There has been also another mail where they have given a link. How does that virtual calculator may work? So numericals will be always there.
Okay, so I think now the doubts are clear. So thank you, Paramita, for clearing the doubts, because I don't get those mails, so I am not sure about those patterns. So yes, so if you have been provided with that information, that's fine. That's great. So without delaying much, let me start today's discussion. So today's discussion, if you have gone through the week 11 contents, then I think you have looked into it, and you have saw that you have seen that it's mostly about the protein engineering. So today we will be discussing about protein engineering. So without delay, I am starting it. In between, if you face any problem, so just stop me and let me know what is your problem in between. Okay. So till now, what we have learned is about the different techniques which help us to identify, or which help us to visualize the three-dimensional structures of the different macromolecules. Mostly, we have talked about protein structure determination, and now we are moving to an advanced field which is emerging very recently, and I think in future it will have a great scope, that is protein engineering. So from the name only, you can infer that what we are talking about here is about we are somehow engineering the protein to serve a specific purpose which we are more interested to, or what I will say, that the function structure function relationship of the proteins that we have discussed previously, so that a structure provides a function. So if you somehow modulate the structure of the protein, then the function will also be modulated, and that people have come across with several approaches how to design the protein so that they work in a more efficient and in a more specific manner. Okay, so as if we want to define protein engineering, it can be defined as improvement in the activity and usefulness of an existing protein, or creation of a new protein function by making suitable changes in its amino acid sequence is called protein engineering. Just like what I said, that you are actually looking into the protein structure, you know about what is its function in our body, and you want to make it more useful, or you want to improve the activity of that protein by making some changes in its primary amino acid sequence, so that it can give you an enhanced function. So the field which is actually performing this is known as protein engineering. And one of the most important protein, if we talk about, is is the enzymes. So the enzymes, they are the driving forces in our body, which are responsible for various processes that are actually very important. This enzymes activity is very important to perpetuate those metabolic processes in our body. So if someone can design a more efficient enzyme which can give you a more specific product in a very small amount of time as well as in a cheap cost, then it has a real significance in the industries as well as in the therapeutics field. There are various fields which can gain advantage from this protein engineering field. So in this slide you can see a picture in your slide which can give you a overview of the protein engineering field. So here there is the parental gene that can you can see in the figure. So that will actually translate what parental gene means: the genes which are available in the nature in our body. Okay, so which are responsible for the physiological functions. So the parental gene can provide you the parental protein, means the physiological protein. But from the physiological protein, what we can understand is that we know its structure, we know its sequence. Then what we can do is we can modulate this function of this parental protein either by two methods which are mostly discussed in sir has discussed in his module, as well as we will find that these two methods have revolutionized the field of protein engineering. One is known as directed evolution, and another, in this figure it is written as computational designing, which is also known as rational designing. So we will talk about each of these designing methods in details in our discussion, in our 2 hours discussion today. Then what in brief what we are doing here is we know the sequence, we know the structure of the proteins. What we do is we introduce point mutations, some mutations in the amino acid sequence of our protein, and then we want to select the best fitted protein which will serve the best function, I would say. So that we will screen out, and that evolved or that protein engineered, that engineered protein will be used for several applications, like that they are very useful in food and detergent industry. They are very useful for gene therapy and various therapeutics interventions. They are very useful in bioremediation, biopolymer productions, and biofuels. So these are some of the coming industries which have shown promising results over these years by applying protein engineering by these two approaches that we will be discussing in more details. Okay. So, before designing something, we need to know about the ABCD of that particular thing that we want to design or we want to go for advanced engineering. So, I would say the ABCD of protein is its primary sequence, or the sequence of amino acids that are fitted with that actually makes peptide bond with one another to form a string of where you can imagine the amino acid a bead, and that is being connected in a string to give a or primary sequence of the amino acids that will actually fold into different secondary structures. Mostly these slides that I am talking about, this I had previously discussed. Since we are again going to protein engineering and sir has discussed this part, so I thought to revise this to get the insights more easily. So from the primary amino acid sequence, the secondary structures, or the secondary structures, mostly the alpha helix and the beta pleated sheets are being made, and these secondary structures are being formed by different
And once these
these
secondary
secondary
structures are
structures
being
are being
formed, they
formed, they
fold
fold forces
several
into
tertiary structures
several
which can
tertiary
provide
structure
you
which
a
can provide you
function.
a
And
function.
these
And
tertiary
these
structures,
tertiary
when
structures,
two or
when
more
two or more
polypeptide
polypeptide
chains
chains they
come
come
together with
together
one
with one
another
another
by
by
different
different
non-covalent
non-covalent
forces,
forces,
they
they
actually
actually
form a
form a
quaternary
quaternary
structure.
structure.
So this
So
is
this is
the
the
basic
basic
outline
outline
of
of
how the
how the
protein
protein
structure
structure predicts or protein structure organization
takes
takes
place.
place.
And
And
now,
now,
just as we
as
were
we were
moving
moving
to the
to the
secondary
secondary
structure,
structure,
so
so
the
the
alpha
alpha
helix.
helix,
So the
so the
alpha
alpha
helix
helix
has,
has,
as
as
we
we
discussed,
discussed,
that
that
there
there
is
is
80
80
N
n
to
to
N
n
plus
plus
4
4
residue
residue
of
of this
carbonyl
carbonyl
carbon
carbon
and the
and the
nitrogen,
nitrogen, which will
the
the
amide
amide
nitrogen
nitrogen
that is
that is
forming the
forming the
backbone.
backbone.
So
So
there is
there
a
is a
hydrogen
hydrogen
bond
bond
between
between
them,
them,
that is
that is
N to
n
N
to n
plus
plus
4
4
hydrogen
hydrogen
bonding
bonding
happens.
happens.
If you
If you
see
see
the
the
means,
means,
if you
if you
see the
see the
axis
axis
of the
of
alpha
alpha
helix,
helix, then the
then the
hydrogen
bonds are
bonds are
perpendicular
perpendicular
to the
to the
axis
axis
of
of the
the helix.
helix.
Okay.
Okay.
And
And this
alpha
alpha
helix
helix
has
have
3.6
3.6
residues
residues
will
will
make
make
one
one
turn.
turn.
So
So
the
the
alpha
alpha
helix
helix
that we
that
are
we are
more
more
right-handed
right-handed
alpha
alpha
helix
helix
that we
that
are
we are
more
more
habituated with.
habituated with.
So
So
the
the
3.6
3.6
residues
residues
make
make
one
one
turn.
turn.
And
And the
from
from
one
one
turn to
turn to
another
another
turn, if
turn,
you
if you
measure the
measure the
distance,
distance,
then
then
it
it
is
is
the
the
pitch,
pitch,
as
as
you
you
can
can
see in
see
the
in the
figure, that
figure,
is
that is
around
around
5.4
5.4
angstrom.
angstrom.
Okay.
Okay.
And the
And the
rise,
rise,
or
or
the
the
rise
rise
per
per
residue
residue
is
is the
if
if
you
you
calculate
calculate
that
that
in
in
that
that
alpha
alpha
helix,
helix,
that is
that
how
how
much
much
distance
distance
one
one
residue
residue
is
is
related to
related to
another,
another,
then you
then
can
you can
calculate
calculate
it
it
as
as
1.5
1.5
angstrom.
angstrom.
Okay.
Okay.
So
So and
what are
what are
the
the
different
different means
helix
helix
formers,
formers,
and
and
what are
what
the
are the
different
different
means different
different
amino
amino
acids
acids
which
which
are
are
less
less
preferred in
preferred
the
in the
alpha
alpha
helix
helix,
is
that is
also
also
listed
listed
in
in
your
your
screen.
screen.
Okay, in the
Okay, in the
alpha.
alpha helix.
Now
Now
there is
there is
another
another
kind of
kind of
secondary
secondary
structures
structures
which
which
are
are which we know, that is the
beta
beta
sheet.
sheet.
And
And
if you
if you
see
see
that there
that
are
there are
two
two
kinds of
kinds of
beta
beta
sheet.
sheet.
One is
One
the
is the
parallel
parallel
beta
beta
sheet,
sheet,
and
and
another one
another one
is
is
known
known
as
as
the
the
anti-parallel
anti-parallel
beta
beta
sheet.
sheet.
Okay.
Okay.
So
So
what
what
in
the
the parallel
in the parallel means
beta
beta
sheets, or
sheets
the
or the
beta
beta
strands.
strands.
So
So
there
there
the
the
hydrogen
hydrogen
bonds
bonds
that
that
you
you
see
see
here,
here,
they are
they are
not
not
coplanar
coplanar
or
or
collinear
collinear
to
to
one
one
another.
another.
But in
But
an
in an
anti-parallel beta
anti-parallel
sheet,
beta sheets,
as
as
there will be
there will
six
be
the
the
hydrogen
hydrogen
bonds
bonds
that
that
you
you
can
can
see in
see
the
in the
next
next
figure, I
figure, I
think.
think. Yes.
So
So
you
you
see the
see the
hydrogen
hydrogen
bonds
bonds
are
are
parallel
parallel
to
to
each
each
other,
other,
and they
and
are
they are
co-linear
co-linear
to
to
each
each
other.
other.
So
So
that
that
makes the
makes the
anti-parallel
anti-parallel
beta
beta
sheets
sheets
more
more
stable
stable
than the
than the
parallel
parallel
beta
beta
sheets.
sheets.
Okay.
Okay.
And
And
also
also
this
these
distance
beta
the
sheets,
distance
the
between
distance
two
between two
adjacent
adjacent
amino acids
amino acids
is
is
3.25
3.25
angstrom.
angstrom.
And
And
typically
typically
beta sheet
beta
has
sheet
several
have several
regions of the
regions of
side
the
chain,
side
and
chain, and
each
each
beta
beta
strand
strand
is
is
around 5
around
to
5
10
to 10
amino acids
amino acids
long.
long.
So
So
another thing
another
that I
thing that
wanted
I
to
wanted to
discuss is
discuss
that
is that
if you
if you
see
see
only
only
the
the
sequence of
sequence
the
of the
amino
amino
acids and
acid and
compare
compare
in case
in
of
case
the
of the
alpha helix
alpha
and
helix
the
and the
beta
beta
strands,
strands,
what it
what
has
it
been
has been
observed that
observed that
in
in
after
after
every
every
fourth,
fourth
third to
third to
fourth
fourth
residue in
residue
an
in an
alpha
alpha
helix,
helix,
it is
it is
either a
either a
hydrophobic
hydrophobic
amino
amino
acid,
acid,
and
and
there is a
there is a
there
or
is a
there is a
hydrophilic
hydrophilic
amino
amino
acid
acid,
which is
which is
actually
actually
repeated
repeated
in a
in a
subsequent
subsequent
fourth
fourth.
after
After
four
four
amino
amino
acids.
acids,
There is
there
some
is some
kind of
kind of
mutation of
reputation
the
of the
nature of
nature
the
of the
amino
amino
acids that
acids
is
that is
present.
present.
But
But
in a
in a
beta
beta
sheet,
sheet,
it is
it is
alternate
alternate
hydrophilic
hydrophobic
amino
amino
acids
acids
are
are
present.
present.
That
That
is
is
actually
actually
an
an
observation
observation
that
that
you
you
can
can
infer
infer
from by
from by looking
looking
into
different
different
sequences
sequences
of
of alpha helix and beta sheet, and that actually helps
you a
you a
lot
lot
while
while
performing
performing
protein
protein
engineering
engineering
or
or
designing
designing
protein.
protein.
So
So
whether
whether
you
you
want
want your means when you are we will discuss about the different modeling techniques where you are doing a de novo protein designing, and you want to have a lot of alpha helix, or you want to have a beta sheet in your protein, then these are the information that will help you to design the protein of your interest by modulating the amino acids and getting a particular secondary structure. So these are all we have discussed previously. And what is a beta pleated sheath? So it is called pleated because the side chains of if you see the side chains of these beta strands which are forming the beta sheet, so the side chains point up and down alternatively. So that's why this is called as beta pleated sheet. Okay, so that's why they are called as pleated. Now, what are the different? So, if we look into a single polypeptide chain, then you will find, as we have also discussed, that the peptide bond, that is the CONH bond. So, due to resonance, it has a partial double bond character. So, since it has a partial double bond character, so if you see, or if you want to rotate this peptide bond in the three-dimensional space, then there are some constraints, and that angle of the peptide bond angle, or the torsion angle, means in the torsion angle or the dihedral angle, as we have discussed previously, that they are the angle between planes. And when how can you define a torsion or dihedral angle? That is, you need at least four atoms to define a particular torsion angle or a dihedral angle. Okay, so whenever that I said you that if someone asks you about a dihedral angle, always mention about the four atoms which are involved in the formation of that angle. So if you see the means the peptide bond, the CONH, if you see this angle, so that is being made by C alpha, CO, the carbonyl carbon, the N nitrogen, the amide nitrogen, and the C alpha of the next amino acid. So these four atoms will make two planes, and the angle between these two planes, that is the angle between this bond, that is the CN bond at the rotation angle. So that is the omega. Okay, so that can take two angles, that is either 0 degree or 180 degree, based on the cis and the trans interaction. So these informations are very important when you want to design a protein de novo, or you want to model a protein. So you need to consider these informations. And I had also previous. I will be coming in the next slide about Ramachandran plot. So whenever you do any designing of your proteins, or means you design or you model a protein, your protein structure should pass through the Ramachandran plot, where it will provide you the allowed regions, whether there is any steric clash is present in your protein structure. If there is any steric clash present in your structure, then that will be not considered as a lower energy structure. Then you have to do multiple steps of refinement to minimize the steric clashes, and to minimize the or lower the energy of your structure. So for that, these informations about these torsional angles, understanding what they mean, is very important. That's why I thought to revise these topics with you a little bit. So then there are this means another. So since the partial double bond character is present in the carbonyl carbon, the peptide bond, so there is restrictions. But this n this C alpha n and this C alpha C, the C means the carbonyl carbon here, and the NC alpha, N is the amine nitrogen, and this C alpha is the C alpha carbon which is present. These two bonds are since they are single bonds, they can easily rotate in the space to take various angles. But however, it has been seen that by G.N. Ramachandran, when he was and his two students, Shashi Shekharan, and what they observed was that although these are single bonds, but they can't take all the angles. If you consider plus 180 to minus 180, if you consider a protein three-dimensional structure, they can't take all the angles. There are certain restraints, or there are certain constraints which are applied because of their orientation in the space. And there are they if they are there are only certain allowed angles which you can see in the Ramachandran plot. So this Ramachandran plot is in the x-axis you have this phi angle that we discussed in the last slide. That is the angle between the C alpha and, or what are the four atoms involved here. The four atoms are the Ci minus 1 means the carbonyl carbon of the previous amino acid. Ni means the amide nitrogen of the current amino acid, C alpha of the current amino acid, and the carbonyl carbon of the current amino acid. So these four atoms, they are actually making two planes, and the angle between them is the phi angle. Similarly, if you look for the psi angle, then this I plus 1 in the, if you see here in the end is the I plus 1 means amino acid, or the amide nitrogen of the next amino acid. Okay, so if you I had showed you in the pi mole as well how to calculate the dihedral angle. So you need to have information about these four atoms for get the dihedral angles. So after getting the dihedral angles of all the proteins, what G.N. Ramachandran theoretically he assumed that means which has been proved means correct by various computational models. In the x-axis is the phi angles, and in the y-axis there are the psi angles. And if you plot, then this region, the blue region, is for the alpha helix, and this red region is for the beta sheet, and this yellow region is for the left-handed, or the green region is for the left-handed alpha helix. Okay, so from this you can see that only the alpha helix, or the right-handed alpha helix, there is certain angles that it can take. Same with beta sheets as well as the left-handed alpha helix. So this is just to show you the different rotations at different angles, at means how this rotation occurs in space. Okay, so we are rotating actually the phi angle here by keeping the psi angle constant. Okay, so now these are the basic understanding about the protein structure that you should have before going into the protein engineering. Since sir always talk about the journey or the history behind the means protein engineering, history behind each field he touches upon. Then so he also taught you about what I have seen, that he also taught you about this journey of protein engineering, means what is the history of protein engineering involved. So I thought that I have not listed the all the seminal contributions, but what I have listed is which I thought that they are more important, or they are more renowned discoveries which actually led means groundbreaking discoveries in the field of protein engineering. So the first one is since we in the first slide also we talked about the different enzymes, means the enzymes which are the driving forces for the for carrying out different metabolic processes in our body. So the enzymes term, or the it was pointed out by Wilhelm Kuhne, and from that
the start of the enzymology, the field of enzymology evolved or started.
and started. And he what he demonstrated is that he demonstrated the cell-free fermentation by the use of this enzyme. So that was the first time when the enzyme term was coined. And later, Edward Buchner, he actually discovered the enzyme that many of you may have known previously. That is the zymase, which he isolated from the yeast that can actually ferment sugar. Okay, so that was also a very important discovery in the field of enzymes. So we I have listed most of, if you see that most of the discoveries I have listed is with enzymes, because means most of the discoveries in protein engineering, if you see that they are more interested to work with different enzymes, because they have much potential, and they have much industrial value, so much application oriented. So that's why the emergence of the field of enzymology in protein engineering is a seminal contribution. So then Michaelis and Menten, they came up with the famous Michaelis and Menten equation that means when we start reading about enzyme kinetics in any biochemistry book, so we come across with Michaelis and Menten equation, which actually tells you about the kinetics of and the kinetics of your reaction, or the enzyme kinetics, how these enzymes are, what is the means catalytic power of your enzyme. All this information we can, what is the maximum rate, what is the Vmax, if you remember, or what is the Km, the Michaelis constant. So all this comes from this Michaelis Menten equation, and that was being provided by Michaelis and Menten. And another important discovery that actually was very important for protein engineering is the discovery of the restriction endonuclease. So if you remember the recombinant DNA technology, which is the work power of the different cloning techniques that is important for designing the proteins in the first hand, to get then further process it for the different structure predictions or protein engineering. So the initial step is the recombinant DNA technology, and there, if you remember, this restriction endonucleases or the restriction enzymes that we use, so they are of much important, and that was given by that was first discovered or characterized by Arber, Nathan, and Smith. Then Hall came up with the mechanism of directed evolution in enzymes. So what is directed evolution? So that we will be coming in few slides, we will be knowing about what is directed evolution. But the first mechanism of directed evolution in enzymes was coined out by Hall. Then Smith came up with targeted mutation in proteins. So means what again means, I will tell you, just wait for two to three slides. We will be talking about also about the targeted mutations that we perform in our lab. Normally, one of them is known as the site-directed mutagenesis, the technique that we perform to provide or to carry out these targeted mutations in the proteins. So that was first proposed by Smith, and then Francis Arnold, he was also awarded the Nobel Prize for coming out with the directed evolution of proteins. So he actually proposed the random mutagenesis. So since I came across this term, so I will tell you that in directed evolution, the random mutagenesis is one of the most important part, I would say, to carry out the directed evolution of the proteins in the protein engineering. So that was actually coined out by Francis Arnold, and for which he and his seminal contributions in the field of directed evolution, he was provided the Nobel Prize. Another important discovery was about the ab initio modeling techniques that I will be discussing with you. Means from how from scratch you can model the proteins nowadays. Means you do not need any previous information about the structure or the sequence of your protein, or but you can get your protein structure, or you can model your protein structure from scratch. So that is the ab initio modeling, and there are two important softwares that sir also mentioned and that we work upon when we work with ab initio modeling. Is one of them is Rosetta, another one is iTaser. So this Rosetta was made by David Baker and his lab members. So David Baker is another scientist who had made seminal discoveries in the field of protein engineering, and he was the first person to actually show us that these proteins, we always used to wonder that the proteins and all these biomacromolecules, they are made by nature, but he first showed us that there is a possibility that where we can actually make a protein in the lab or in computer starting from the scratch. Okay, so and he designed the software Rosetta, and I am sure that sir also mentioned that in future he may also get a Nobel Prize because of his seminal contribution in the field of rational designing or computational modeling of proteins. Then he also did the computational enzyme design to engineer Kemp eliminase to erythroaldolase. Then came George Smith and Winter, who also won a Nobel Prize for fast display. So the fast display, in short, I would say that it is a technique which is being used in case of directed evolution. So when we will discuss about the directed evolution, you will see that in case of protein engineering, that I in the first slide, if you remember, what we do is that we do some mutations into our proteins, then we select the best the proteins on the basis of its function. So if it's an enzyme, then we will check the enzyme activity. But if we are interested in finding the binding partners, or we are more interested in the ligand receptor docking or ligand receptor interactions, then this fast display is a very important technique in that part, where you are making the mutant proteins, and those mutant genes that will synthesize the mutant proteins, those mutant genes are being integrated into a fast genome. And in the fast genome, they are integrated at the positions where those positions of the gene, they are responsible for the expression of their surface proteins. Okay, so when these phages they actually expresses the surface proteins, they also display the mutant protein of interest that you have, or the gene of the mutants that you have incorporated into the phage, and that will display your protein. And that's why it's known as the phage display. And in that you can actually in a single time you can check the interaction. If you are interested in finding the binding partner of your protein, or you are looking to increase the binding strength of a ligand and a receptor, then with this fast display you can screen out those mutants which can bind more strongly with your protein of interest. And so in that way, this is a selection. This has given us a technique through which we can select our protein of interest in the protein engineering field, particularly in the directed evolution of the proteins. So that's why Smith and Winter, they were also provided with the Nobel Prize for coming up with this excellent discovery of fudge display. Another means discovery that I think most of you know, that is AlphaFold, and the current version is the AlphaFold 2. So that AlphaFold is a contribution of all these discoveries that has led to this computational, I would say, paradigm. So which actually has shifted the field of structural biology in a different level, and the product of so much investment in this protein engineering field is the AlphaFold 2. So that so what are the different, so this is a gist about the journey of protein engineering. And now if we as I had said multiple times also in the previous been slides, so if we want to divide the protein engineering field into two major classes, then it will be rational design and another is directed evolution. So what is rational design? Rational design needs some prior information about the three-dimensional structure of your protein, as well as what is the function it performs. So these two information, you need to have certain previous information about the three-dimensional structure, that is that one needs to know before starting the rational design. Okay, so why, we will be coming in the next slide. And if we talk about directed evolution, then it is somehow, I will say, that we are trying to, that directed evolution is a quite old technique and rational design is a new technique as we compare to the directed evolution. Okay, so if I will say that directed evolution, when previously the scientists thought of, they tried to replicate what is being done in nature. Okay, so how these different mutations come up in the nature, or how these proteins evolve to provide the best function, and how these proteins which provide the best functions are being selected, that actually scientists used to wonder, and they came up with the field of directed evolution. So directed evolution of proteins is a process of mutation and selection. So you will see that what we do is that we have a original protein sequence that is present with us, and we actually do random mutagenesis into the protein sequence, and we actually select those mutants that I was talking about in the first display, which actually gives the best function. And so that's what it is written, that directed evolution of proteins is a process of mutation and selection, whereby the encoding DNA, the DNA which is coding for the parental protein or the original protein, is mutated, and the resulting variants are screened and selected for specific functionality. So if we in a nutshell, if we want to if we want to compare between these two techniques of protein designing, that is the rational design and the directed evolution, so I have also provided some links in the reference section in the below in below of the slide, then which you can actually refer, or there are some papers and some book chapters that I had followed while making the slides. So I will also share with you these book chapters or the papers that I had followed while making the slides in the means in the Google Drive. So there also you can access those. And if you are interested in this field of protein engineering, you can definitely go through them and get some more details about these two techniques. But in gist, if I want to compare between rational design and directed evolution, then in the left you can see that first is the rational design, where where you have a wild type gene or a parental gene,
and
and
you
you
have
have
to
to
come
come
up
up
with
with
a
a
structural
structural
model,
model,
okay,
okay,
by
by
doing
doing
some
some
different
different
I
I
have
had
talked
talked
with
with
you
you
about the
about the
simulations,
simulations,
about the
about the
different
different,
there are
there are
different
different
computational
computational
tools,
tools,
there are
there are
different
different protein
modeling
modeling
tools
tools
with
with
which
which
you
you
have
have
to
to
come
come
up
up
with
with
the
the
structure of
structure
your
of
protein
your
that you
protein
want
that you
to
want to
actually
actually means
engineer.
engineer.
And
And
if
if
there are
there are
any
any
experimental
experimental
structures that
structures that
is
is been
present
present,
and
and
you are
you are
working with
working
a
with a
protein
protein
which is
which is
whose
whose
experimental
experimental
structure is
structure is
not
not
present,
present,
but
but
the
the his
that
that
protein
proteins
that protein analog homologous
homologous
structures are
structures are
present,
present,
then you
then
can
you can
actually
actually
model
model
them
them
with the
with the
different
different
modeling
modeling
techniques
techniques
that
that
I will
I
be
will be
talking
talking
about.
about.
So the
So the
first
first
and the
and the
foremost
foremost
thing
thing
is
is
that
that
you
you
need to
need to
have a
have a
structure,
structure,
and
and
you
you
need to
need to
know
know
that
that
which
which
sequence
sequence
is
is
providing
providing
you
you
a
a
particular
particular
which
which
structure is
structure
being
is being
provided by
provided by
which
which
sequence.
sequence.
The
The
parental
parental
gene
gene
will
will
provide
provide
you
you
what
what
type of
type of
three-dimensional
three-dimensional
structure in
structure
the
in the
space
space
that
that
you
you
need
need
to
to
know
know
previously
previously
for the
for the
rational
rational
design.
designing.
Then
Then
after
after
since
since
you
you
know
know
about the
about the
structure
structure
as well
as
as
well
the
as the
function
function
previously
previously
with
with these
different
different
computational
computational
modeling
modeling
tools
tools,
as
as
well
well
as
as the
different
different
structure
structure
determination
determination
techniques that
techniques
we
that
have
we have
talked
talked
about,
about,
now
now
you
you
know
know
that
that
in
in
which
which
region
region
you
you
want to
want to
mutate,
mutate,
and that
and
can
that
give
can
you
give
a
you a
better
better
function,
function,
because
because
since you
since you
know the
know the
structure you
structure, you
also
also
know the
know the
function of
function of
your
your parental
protein.
protein.
Okay,
Okay,
so
so
now
now
you
you
can
can
design
design
very
very
specific
specific
mutations
mutations
which
which
can
can
increase
increase
the
the
processivity
processivity,
or
or
which
which
can
can
increase the
increase the
functionality
functionality
of
of
your
your
protein.
protein.
Okay,
Okay,
so
so
that's
that's
why
why
we
we
here
here
perform
perform
site-directed
site-directed
mutagenesis.
mutagenesis.
Means
Means
we
we
want
want
to
to
see
see
after
after
mutating
mutating
that
that
particular
particular
residue
residue
which
which
was
was
actually
actually
important
important
for
for
the
the
function
function
of that
of that
protein,
protein
the
in the
parental
parental
protein,
protein,
whether
whether
mutating
mutating
that
that
mutating
mutating
that
that
residue
residue
or
or
that
that
amino
amino
acid
acid
is
is
somehow
somehow
increasing
increasing
its
its
function
function
or
or
not.
not.
Sometimes we
Sometimes we
can
can
also
also
do
do
this
this
site-directed
site-directed
mutagenesis
mutagenesis
where we
where we
want
want to
to see
see
whether the
whether the
function
function
is
is
decreased
decreased
or
or
not.
not.
So
So so
we
we
will
will
perform
perform the
site-directed
site-directed
mutagenesis.
mutagenesis.
So
So
what is
what is
site-directed
site-directed
mutagenesis
mutagenesis,
that
that
we
we
will
will
also
also
discuss
discuss
in
in
one of our
one of our
later slides.
later slides.
So once
So
you
once
have
you have
done that
done that
site-directed
site-directed
mutagenesis,
mutagenesis,
you
you will be
creating
creating
that
that
mutants
mutants
in
in
your
your
actually
actually
what we
what we
do
do
is
is
that
that
we
we
get
get
that
that
mutant
mutant
gene,
gene,
we
we
transform it
transform it
into
into
bacterial
means
bacterial
bacterial
strain
strain.
for
For
mostly
mostly
we
we
use
use
E.
E.
coli
coli
DH5
DH5
alpha,
alpha,
where
where
we
we
transform
transform
these
these
mutant
mutant
genes
genes,
and
and
then
then
we
we
express
express
our
our
proteins
proteins,
or the
or the over
overexpression of
expression of our
proteins we
proteins
will
we
do,
will do.
and
And
then
then
we
we
will
will
actually
actually
subject
subject
it
it
with
with
certain
certain
selection
selection
criteria
criteria,
where we
where
will
we will
be
be
if
if
it's an
it's an
enzyme
enzyme
we are
we are
interested on,
interested on,
we
we
will
will
look
look
into the
into the
enzyme
enzyme
activity,
activity.
or if
Or
it's
if it's
we
we
are
are
more
more
interested
interested
in the
into the
ligand
ligand
receptor
receptor
interactions,
interactions,
then we
then we
will be
will be
looking
looking
into the
into the
interaction
interaction
strength
strength
of
of these
mutant
mutant
genes,
proteins.
and
And
then
then
we
we
will
will
actually
actually
try to
try to
design
design
these
these
novel
novel
proteins
proteins
for
for
performing
performing
more
more
specific
specific
functions.
functions.
Okay,
Okay,
so
so
that is
that is
about
about
this
this
rational
rational
designing.
designing.
But
But
what we
what we
do in
do
a
in a
directed
directed
evolution
evolution
is
is
that
that
we
we
have
have
a
a
parental
parental
gene,
gene,
and
and
we
we
don't
don't
have
have
that
that
previous
previous
information
informations
about,
about,
or we
or we
don't
don't
require
require
any
any
previous
previous
information
information
about the
about the
structures
structures
at the
at the
first
first,
or
or
when we
when
are
we are
starting.
starting.
So
So
what we
what
do
we do
is we
is we
take the
take the
parental
parental
gene
gene,
and
and
we
we
introduce
introduce
random
random
mutations
mutations
in
in
that
that
parental
parental
gene.
gene.
And
And
then
then
again,
again, means
how we
how
do
we do
that
that,
is
we do
that
that
by
by means you
we will
will
see
see
that
that we will
discuss
discuss
that
that
together the random
random
mutagenesis
mutagenesis
will be
will be
happening
happening,
means
means
not by
not
the
by the
side
side-directed directed mutagenesis protocol,
that
but
there
there
will
will
be
be
point
point
mutations
mutations
throughout
throughout
the
the
genome,
genome, which will make
a
a
certain
certain
random
random
mutagenesis,
mutagenesis.
or
Or
we
we
can
can also means
in
in
that
that
in
in
this
this
field
field
of
of directed
evolution,
evolution,
what
what
people
people
have
have
found
found
that
that means
they
they
can
can
also
also
carry
carry
out
out
certain
in different recombination techniques. One is known as this DNA shuffling. If you remember, sir talked about in his slide there, where you take different the DNA or the genes of which are more related to one another but for different organisms, and then you actually do the recombination. That actually gives you an advantage of making the mutations in a more productive way. I would say that what the proteins that you will be getting after making those mutations, they are more means that you need to actually process by different screening criteria. So first you make the random mutagenesis and the recombinations, and you take that mutants or the recombinant gene and transform it again into an E. coli, or in an a transform it into an E. coli strain. Okay, so then what you do is that you again purify, or you express your protein, you purify your proteins, and then you carry out a high throughput screening of the functionality of the proteins. The proteins which are more favorable for your functions, they are being selected, and their genes are again again going through the same iterative process of directed evolution, uh, uh, till that level where you are getting the best function of your protein. So you get a protein which is more evolved, much evolved than the previous one, that is giving you a positive selection. You choose those population of the genes or the mutants, and then again do the similar directed evolution process to get a much more advanced protein of which will have a more advanced function than the previous one. So in this way you will carry out this iteration till that point you to which you get a the best functionality of your protein. So that is the way of directed evolution. So if you this is for just for these two tables are just for your means knowledge, means I would say how to compare or what are the things that is required for carrying out these two techniques. So like for knowledge of the protein structure, for rational design it's required, but for directed evolution, as we are carrying out random mutagenesis, we don't require this knowledge of the protein structure at the start. Okay, and then means there are different points that you can go through, that what are the differences between this rational and rational design and the directed evolution. Okay, now we are moving more into the rational protein design. So if we again divide this rational protein design into some categories, then we can divide it into three major categories. First is completely novel protein design, which I would say this is the de novo protein design that you can do with the help of this rational protein designing. De novo means from the scratch you are designing a protein, means there is no prior informations which are available, or there are no protein structures which are available, but you are engineering your protein in a de novo fashion. So that can comes from the completely novel protein design, then blob level protein design, and the protein variants designing. Okay, so the completely novel protein design, so there is no prior experimental information which is being present in this case. The protein could be designed ab initio on a single computer. Ab initio means starting from the scratch into means there are how these ab initio methods are being carried out, that we will be also talking in the next slides. Then we can take the help of the protein structure prediction algorithms, means there are certain theoretical algorithms that you will be been seeing you will be experiencing in the next few slides, where we will be talking about the different energy algorithms, or there are different constants which are being made in computationally that help us to predict the protein structure with these different theoretical algorithms. Okay,
then
then
we
we
can
can
also
also
here
here
take the
take the
help
help
of
of
protein
protein
structure
structure
fold
fold
prediction
prediction
using the
using the
approach
approach
of
of
machine
machine
learning.
learning.
So
So
this
this
machine
machine
learning,
learning,
as
as
we
we
all know,
all know,
is
this is
a
a
data
data
driven
driven
process
process
where
where
we
we
actually
actually
take
take
the
the
help
help
of
of
several
several
statistical
statistical means
methods
methods,
and
and
also the
also the
data
data
which is
which is being
created
created
previously
previously
with
with
the
the
structures
structures
which are
which are
actually
actually
available
available
in the
in the PDB
or
or
in the
in the
different
different
databases.
databases.
And
And
if
if
we
we
have,
have,
when
when
you
you will
see
see
that
that
we
we
will be
will be
talking
talking
about the
about the
different
different
modeling
modeling
techniques,
techniques,
then you
then
will
you will
see
see
that
that
we
we
make
make
use of
use
those
of those
informations
informations
which are
which are
previously
previously
available
available
in
in
machine
machine
learning
learning
to
to
actually
actually
solve
solve
or
or
design
design
the
the
protein
protein
structure.
structure.
Okay.
Okay.
Now,
Now, then the
another
another
level
level
of
of
protein
protein
design,
design,
of
of
rational
rational
protein
protein
design, I
design,
would
I would
say
say,
that
that
is
is
the
the
blob
blob
level
level
protein
protein
design.
design.
So the
So the
basic
basic
idea is
idea is
to
to
here
here
to
to
combine the
combine the
protein
protein
units
units
of
of
defined
defined
functions. So
functions. So
here
here
you
you
know
know
about
about
the
the
functions
functions
of
of
particular
particular
domain,
domains,
and
and
you
you
want to
want to
combine
combine
two or
two
more
or more
domains to
domains to
give
give
a
a
novel
novel
function
function,
or to
or to
give a
give a
more
more
efficient
efficient
function
function
of
to a
protein.
protein.
So what
So
we
what
do,
we
we
do, we
take two
take
or
two
three
or three
domains
domains
whose
whose
functions
functions
we
we
already
already
know.
know.
So what
So
are
what are
domains? They
domain,
can
they can
independently
independently
fold
fold
in the
in the
three-dimensional
three-dimensional
space
space
to
to
provide a
provide a
function.
function.
So
So
that's why
that's
we
why we
know the
know the
function
function
of
of
each of
each
the
of the
domains,
domains,
and we are
and we are
connecting those
connecting those
domains with
domains
the
with the
help of
help of
certain
certain
linker
linker
regions.
regions.
Okay,
Okay,
and
and
making
making
a
a
complex
complex
of
of
different
different
domains
domains
that will
that will
perform
perform
a
a
function.
function.
Okay,
Okay,
so
so
that is
that is
the
the
blob
blob
level
level
protein
protein
design.
design.
So
So
what you
what you
require
require
in a
in a
blob
blob
level
level
protein
protein
design
design
is
is
the
the
rough
rough
geometry
geometry
of the
of the
protein
protein
domains
domains
that you
that you
need to
need to
know.
know.
Okay,
Okay,
so
so
it
if
may
it
be
may
some
be
low
some low
resolution
resolution
structures
structures
that will
that will
also
also
do,
do,
but
but
you
you
should be
should be
able to
able
know
to
the
know the
rough
rough
geometry of
geometry
your
of your
protein
protein
domains.
domains.
What are the
What are the
secondary
secondary
structure
structure
if
if
insertions or
insertions or
disruptions are
disruptions are
planned?
planned?
So if
So
you
if you
are
are
what
means
are
what are
insertions or
insertions or
disruptions?
disruptions?
So
So they, while
doing
doing
some
some
mutations
mutations
in the
in the
protein
protein
designing,
designing,
if you
if you
want to
want to
insert
insert
a
a
particular
particular
amino
amino
acid or
acid, or
insert
insert
a
a
particular
particular
tag
tag
sometime
sometime
in the
in the
protein,
proteins,
then you
then
need
you
to
need
know
to know
also
also
about the
about the
different
different
secondary
secondary
structures
structures
that
which are
involved
involved
in
in
that
that
form,
form, involved in
formation
formation
of
of
these
these
different
different
domains.
domains.
And then
And then
also
also
the
the one of the
most
most
important
important
part
part
of
of
this
this
blob
blob
level
level
protein
protein
design
design
is
is
to
to
choose
choose
a
a
correct
correct
linker
linker
region.
region.
So
So
sometimes
sometimes
what you
what you
want
want
is
is
that
that
your
your
linker
linker
region
region
should
should
be
be
very
very
flexible,
flexible,
because
because
if it
if
is
it is
more
more
rigid,
rigid,
then
then
the
the
flexibility
flexibility
of the
of the
two
two
domains
domains
is
is
actually
actually
hampered.
hampered.
So what
So
you
what you
see
see
is
is
that
that in a
three-dimensional
three-dimensional
form,
form,
when they
when they
actually
actually
perform the
perform the
function,
function,
each
each
domain,
domains,
they will
they
have
will
a
have a
particular
particular
orientation
orientation with
which
which it
interacts with
interacts
its
with its
ligand to
ligand and
perform
perform
its
its
activity.
activity.
So
So
if
if
the
the
ligand is
ligand
too
is too
rigid,
rigid,
then
then
it
it
may
may
hamper the
hamper the
flexibility
flexibility
or
or
the
the
conformational
conformational
orientation of
orientation
these
of
two
these two
domains,
domains,
which can
which can
actually
actually
hamper the
hamper the
functionality
functionality
of
of
your
your
protein.
protein.
So
So
in
in
that
that
case,
case, so
this
this
normally
normally
what
what
people
people
do
do
is
is
that
that they
use the
use a
linker
linker
region
region
which
which
is
is
more
more
glycine
glycine
and
and
proline
proline
rich.
rich.
Why
Why
glycine and
lysine and
proline
proline
rich?
rich?
Because
Because
glycine,
glycine,
as you
as you
know,
know,
that
that
they
they
are
are
most
most
flexible
flexible
amino
amino
acids
acids
among
among
the
the
20
20
amino
amino
acids we
acids we
know.
know.
And
And
proline
proline
can,
can,
due
due
to
to
its
its
cis
cis
bond
bond
formation,
formation,
and it
it
can make
can
sharp
make
turns.
sharp turns.
So
So
these two,
these
when
two,
we
when we
use
use
combinations
combinations
of
of
these
these
two
two
amino
amino
acids to
acids to
form the
form the
linker,
linker,
it can
it can
provide
provide
a
a
more
more
flexibility
flexibility into the linker region of the two domains. Okay, so one of the means example of this blob level protein design is the nanobodies. So the nanobodies is that we know that these antibodies, they have this Fab and this FC regions, and in the Fab region there is this variable region as well as the variable region as well as the constant regions are present, but the variable regions of both the heavy chain and the light chains, they are actually responsible for interacting with the antigens in an antibody. But what people have found is that as you know that if we want to administer these antibodies in a patient's body, the lower the molecular weight, the more easily it can be administered into a patient, or it can also reduce the cost of the protein. It can make it more stable. Okay, so we have the information of the antibodies, but now what people have come up that if we only have it's from llama, as I remember that from that they actually first discovered these nanobodies. Okay, so from a kind of from the in the camel also you will find this kind of means nanobodies which is present, where only the variable domain of your heavy chain is actually sufficient to interact with your antigens. So people have observed that, and actually they utilize that information in creating these nanobodies, where this you have the variable region of your heavy chain, and then if you want that there you want to design a complex of antibody which can recognize a wide variety of antigens, then you can do this kind of clusterings as you see in the figure. So if you design a biotin tagged nanobody here, and as you know that this biotin can interact with streptavidin, the streptavidin one streptavidin can interact with four biotins at a time. So if you have a biotinylated end of in the nanobody, then four nanobodies can actually interact in clusters that will also increase its affinity as well as avidity, and also serve various purposes. If you have variable heavy regions of different antibodies clustered into a single complex, so that can be also done. So there are bispecific and nanobodies, there can be single chain nanobodies. Here all the means chains they are coming from a single antibody, whereas there are two chains which are coming from two different antibodies. That can be also possible, which is known as the bispecific nanobody formation. Okay, now so now, since we talked about that, the for the rational designing, what is the foremost thing is to you need to get a structure. Okay, so that you may not have that opportunity to perform means experimental methods there. You can also do the modeling. So that's the beauty of all these contributions of protein engineering or the computational advancements. So what how these protein structure and prediction methods can be classified? They can be classified into three categories. So the first one is knowledge based methods, the second one is energy based methods, and the third one is the ab initio methods or the ab initio modeling. The knowledge based methods means from the name only you can tell that we are actually more relying on certain knowledge of or previous knowledge of some structures which whose structures are already been solved and deposited in some database like pdb. So they are also these knowledge based methods can be also divided into two subdivisions. So one is homology modeling, another one is fold recognition or threading. Okay, homology modeling means there is a homologous structure, at least 60% identical or similar structure is available in the database or the protein data bank, which is more similar or homologous to your protein of interest. Then you can use that structure as a template, and there are different homology modeling tools which will actually use that structure as a template and predict your protein of interest structure, and that is a kind of prediction, but for that you need to have a homologous structure available in your database. Another one is the fold recognition or threading that we will be going deeper into the next slide. In the next slide, and there is also some methods which are based on some energy minimization and the ab initio methods, where you find the solution of a protein folding problem in a conformational space. So what is ab initio modeling, that we will discuss in the next slide. Another important thing that I wanted to discuss with you, there is a competition that always goes on. I think after 2 years, the name of this is the critical assessment of protein structure prediction. That is the CASP. Okay, so in this is a competition where from worldwide various computational biologists they come, and they come with their own algorithms where they can actually predict the protein structures, okay, in a more specific or in a more correct way. So I would say that this competition has made actually seminal contributions to the discoveries of protein engineering. And AlphaFold 2 is also means the AlphaFold 2 Google's DeepMind's AlphaFold 2, they were the winner of this CASP event in 2020. So AlphaFold 2 has also emerged from this competition. Another two important softwares which has revolutionized protein, specifically this ab initio protein modeling, is this iTracer and Rosetta. Okay, so so now let us just as in the previous slide we were discussing about the fold recognition or the threading process. So here what is done is in molecular biology, protein threading, also known as fold recognition, is a method of protein modeling which is used to model those proteins which have the same fold as proteins of known structures but do not have homologous proteins with known structure. Means this fold prediction or threading can be only used when means people use this only when there is no homologous structure which is present. Okay, so as you see that there are many computational power involved in this process, so it is much more costlier than those which already have these homology models are present or homologous structure is present. So first you should look whether there is some homologous structure already present in your protein data bank. If not, then people go for this fold recognition or threading, as people have seen that those structures which are being deposited in the PDB, they have a particular pattern in their folding. There are particular folds which is being repeated continuously to make these three-dimensional structures. So you have that information in the PDB. So from those information of the folds, different folds, this fold recognition or threading process is governed. Okay. So what is threading? Threading means placing and aligning. So what is placing and aligning? Each amino acid in your target sequence means the sequence that you want to model. So the each amino acid in your target sequence to a position in the template structure. So what you do is you are placing and aligning each amino acid of your target sequence with the position or with that position in the template structure. Okay, so in the template structure we'll also have a sequence. You are actually placing and aligning those amino acids in your template structure. Okay, and evaluating that those folds, the template structures are coming from the different folds which are already known, and evaluating how well the target fits to the template. Okay, how well your target fits to the template of a already known fold. Okay, after the best fit template is selected, after the best fit template, which is there are different scoring patterns that you need to create, that is based on different other conditions. Okay, environmental conditions, energy minimizations, and various parameters that you need to consider to define a scoring function. So that scoring function will tell you that how much more aligned or they are more related, more closely related your target sequence and your template sequence is. And on the basis of that you choose a template, and the template is selected, and the structural model of the sequence is built based on the alignment with the chosen template. So based on the chosen template, you are making a best model which is energy minimized, and also all the restraints that are present that can hamper the protein folding process, they are all taken care of by this pouring function that you define. Okay, so what are the major steps? So the construction of a structured template database, where you need to have a number of templates with which you want to align your target sequence. Then what you need to do is you need to design a scoring function. Then we do this threading, that is the we take the amino acids and we align it into the template structure and see the scoring function which will give us a best fitted model, and then we do the prediction of our three-dimensional structure. So these steps have been already followed by many computational biologists, and they came up with various fold recognition or threading softwares. Some of them I have actually listed in this slide below. That is AGHFred, Raptor, and FIDE. Okay, so these are some of the means softwares that one can use for this fold recognition and threading process. Now another one is this, the another more important that I would say, that people are now working with just modeling the proteins from the scratch, that is about the de novo protein designing, that is through the ab initio modeling. Okay, so what it requires is a free energy function. So these energy functions, they are being derived. There are two different methods. One is physics based, another one is knowledge based. Okay, so in the physics based, where you are coming up with the different energy functions, I am giving you an overview or the summary of this modeling, but it is a much means demanding technique, I would say. I will suggest you to go through a material that I will share with you, that is more concentrated on this ab initio modeling. If you are interested to work more on this ab initio modeling techniques in future, that will actually help you. So that I will provide you in the Google Drive. So, but in summary, what you require is a energy function. Okay, here you are not, it's not important that you will have a previous structure or sequence information that is important in ab initio modeling. What it requires is to set up an energy function that is either physics based or some means physics based, means there are certain forces which we need to consider, like the electrostatic forces as well as the coulombic forces and the quantum mechanics, which is actually involved in the formation of the bonds between two atoms. So we know that these amino acids are made up of certain atoms, so those atoms can be bonded in a certain angle. So those things, information we know, then only we can actually do this physics based energy function defining. Or what people do is that already the means the energy functions which are being used for the previously ab initio modeling patterns, or the structures which are already solved, there you have some means energy functions which are being already defined for those structures. So from those energy functions, we are taking or deriving those energy functions and utilizing that in your modeling, and seeing whether that energy function is actually working fine for your protein or not. So that is knowledge based making of your energy functions. And then what you know, what you need is a search tool for your conformational space. Okay, a searching tool for your conformational space that you also require for this ab initio modeling. So you have a target sequence, you need to have your potential or the energy function on the basis of which you are actually means designing your bits or modeling your protein. So on the basis of that energy function, your sequence of amino acids or the target amino acid will be taking a three-dimensional configuration in the space in under this energy function. and then you have this Monte Carlo fragment assembly, where now first you have to see the backbone, whether the backbone of your proteins are energy minimized. Then for conformational space means how your side chains conformations are responsible for means giving you a low energy structure, that actually can be done by choosing a different conformational space for your particular protein of interest. From that you actually get low resolution models, and after defining your models with several parameters which are physics based atomic refinements, you do to perform to get a final atomic model. So what is the advantage? Advantage is to get a fold which is quite novel, that is not previously solved or previously known by the structural biologists, that you can do with this ab initio modeling. And the disadvantage is it can now means, since you see that here there is a lot of theoretical knowledge is important here, and you are not giving your models based on some previous structures which are present. So that's why this ab initio modeling has been successful for only short sequences till now. Okay, so but people are working on it how to extend it to larger sequences. Okay, so the success you can see that in the AlphaFold 2. So where you just give the sequence of your amino acid, now you can get a prediction model of your the model which is quite accurate with the experimental structures that is available for that protein. So from what are the two major methods for ab initio modeling? They are the iTaser and the Rosetta. Okay, so this is just an overview of how this iTaser works. So Rosetta also works in a similar manner, where you get the information about, or where you can model the three-dimensional structure of your protein in Rosetta as well. But the Rosetta has its different energy functions as well as different conformational space restraints that I talked about in the initial ab initio modeling. So what we do here in iTaser is that you have the sequence that you want to model, then you actually perform here a threading process, where where you want to choose a template which is present, which you will be using for making a novel fold. Means there can be multiple folds which are present, but from the sequences you perform a threading just like fold recognition and threading, and then you choose a template, and those templates are being means the best fitted template, you choose it, and you then fragment your template, and then in an energy function you apply an energy function and let this template to reorganize into different clusters, so which is known as the clustered centroid. And these cluster centroid are actually the lower energy structures which are also again they are being subjected to for reassembly, and further there will be again in energy minimization of these structures, and ultimately you will get a final model. So it is just a overview of but how iTaser works. But if you want to read in details, I will share the material with you. So where I was mentioning that it is about ab initio modeling. So there you can get information about more about iTaser as well as Rosetta in that material as well. Okay. Now, so till now what we have discussed is how to get the the first step in the rational designing is to get the three-dimensional model of your protein. So how to get that we have discussed thoroughly about the, or I would say in summary about the different techniques which are available for that. Now the next step in rational designing is to do a targeted mutagenesis. So what kind of mutagenesis is performed? So either there can be the mutagenesis, either there can be point mutation. Point mutation means at a single nucleotide level, or at a single amino acid level you are doing the mutation. Okay. Large modifications means you are mutating the protein in such a way that it's completely is changing, means at each and every level you are doing the mutation. Yeah, you can imagine that you are doing point mutations throughout the length of your sequence, then it's a large modifications that can happen due to that mutations. Okay. So what are the different kinds of point mutations? So if we divide the point mutations, then they can be substitution, they can be insertion, or they can be deletion. Okay. So what is substitution? Substitution means there is only change of one nucleotide. You are actually substituting. So from the word only you can be able to know what it means. Is that you are substituting one nucleotide with another. You are changing A with another, but you are not adding or deleting something in the sequence. You are just substituting with one nucleotide, then it is known as substitution. Now, insertion. Insertion, from the name itself, you are inserting, means you are adding some additional nucleotide in your sequence. Okay, you are adding a one additional nucleotide in your sequence, or you are inserting an additional nucleotide in your sequence, that is known as insertion. Deletion means you are deleting one nucleotide from your sequence. So these are the different ways through which you can do the point mutations. Okay, so there are different types of point mutations. So they can be silent based on the bits function that it provides. It can be divided into different types, the point mutations. One is, if you see this example in the lower panel, it will be clear to you. So there is no mutation. So this codon is in the mRNA level as well as in the protein level. So they are given in three categories. So this codon actually in the DNA level, they code for an amino acid lysine. So if there is no mutation, then it will be coding this amino acid. The tRNA will come and it will read this codon in the mRNA, and it will code for lysine. Now, if you make a mutation in the DNA level, for example, in the TTC you make it TTT, and it makes it into AAA. Now you know that there are different degenerate codons which are present, which will code for a single amino acid. So somehow you made the mutation, but the codon which resulted because of that single point mutation, or that substitution, here they are substituting with from T with C, bin C with T, then we found a codon which codes for only lysine only. Then there is no change in the function, or no change in the translation process. Okay, this will code only for lysine. So that's why this mutation is known as a silent mutation. Means this is silent. It is not hampering something in your protein level. Okay, you are doing the mutation, still you are getting the same amino acid. So that these kind of mutations are silent mutation. What is nonsense mutation? Again, from the name you can only see that how when you tell nonsense, means it's not making any sense here. Means it's not making any sense, means it's not coding for any amino acid. So that it can code for a stop codon then. So where your amino acid synthesis will halt. So when your mutation is actually leading to a stop codon, then it is the nonsense mutation. Now, a missense mutation, that can be conservative as well as non-conservative. Missense mutation means there is a mutation, but it can be conservative, means lysine is a positively charged amino acid. So you are performing a mutation such that it is converting into an arginine. It is converting into an codon which is coding for arginine, but their nature is same. Nature by the means I say that both are positively charged amino acid.
So
So
that's
that's
why
why
there
there
is
is
not
not
that
that
drastic
drastic
mutation
mutation
happening
happening
here.
here.
So
So
it is
it is
conserving,
conservating,
it is
it is
conserving
conserving
its
its
nature
nature
here. So
here. So
that's
that's
why
why
it's a
it's a
conservative
conservative
missense
missense
mutation. Mutation.
mutation.
Non-conservative
Non-conservative
means
means
you
you
are
are
mutating
mutating
with
with
threonine.
threonine.
So
So
threonine is
threonine
a
is a
polar
polar
amino
amino
acid,
acid,
but
but
it's
it's
not
not
a
a
charged
charged
amino
amino
acid,
acid,
positively
positively
charged
charged
amino
amino
acid
acid
like
like
arginine
arginine
or
or
lysine.
lysine.
So
So
here
here
you are
you are
mutating
mutating
that
that
amino
amino
acid
acid to
to some
some
other
other
amino
amino
acid
acid
with
with
a
a
different
different
nature.
nature.
So
So
here you
here
are
you are
not
not
conserving
conserving
its
its
nature.
nature.
That's
That's
why
why
it's
it's
non-conservative
non-conservative
missense
missense
mutation.
mutations.
So
So
these are
these
the
are the
different
different
point
point
mutations.
mutations.
Another
Another
point
point
mutation
mutations
is
is
the
the
frame
frame
shift
shift
mutation.
mutation.
So the
So the
frame
frame
shift
shift
mutation,
mutation,
as you
as
can
you can
see
see
here,
here,
is
is
that
that
you
you
have
have
a
a
wild
wild
type
type
sequence,
sequence,
which is
which is
perfectly
perfectly
coding
coding
for
for
this
a
particular
particular
amino
amino
acid
acid
sequence,
sequence,
and there
and
is
there
a
is a
stop
stop
codon
codon
where
where
there
there
will
will
be
be
a
a
halt
halt
in the
in the
translation
translation
process.
process.
But
But
what you
what
are
you are
doing
doing
here
here
is
is
that
that
you
you
are
are
adding
adding
or
or
deleting
deleting
certain
certain
nucleotides
nucleotides
from your
from your
sequence.
sequence.
So you
So you
know
know
that
that
these
these
amino
amino
acids
acids
are
are
read
read
in
in
triplets,
triplets,
right?
right?
From
From
the,
the,
if
if
you
you
see the
see the
five
5
prime
prime
and
end
the
to the
three
3
prime end,
prime end,
then
then
this
this
all
all these
amino
amino
acids
acids
are
are
read
read
in
in means
all
all these
nucleotides
nucleotides
are
are
read
read
in
in
triplets,
triplets,
and
and
each
each
triplet
triplet
will
will
give
give
rise to
rise
an
to an
amino acid.
amino acid.
Suppose
Suppose
in
in
this
this
sequence
sequence
you are
you are
deleting
deleting
one of
one
the
of the
uracil
uracil
from in
from
this
in this
wild type
wild type
sequence.
sequence.
Then
Then
what will
what will
happen
happen
is,
is,
since
since
this
this
one of
one
the
of the
amino
amino
acid is
acid is
gone,
gone,
so
so
till
till
this
this
the
the
reading
reading
of the
of the
tRNA will
tRNA
be
will be
fine
fine
because it
because
will
it will
read
read
333.
333.
Since in
Since
this
in this
sequence
sequence
one
one
uracil is
uracil is
absent,
absent,
now
now
what it
what
will
it
read
will read
here
here
is
is
that
that
it will
it will
start
start
from a
from a
different
different
nucleotide
nucleotide,
and
and
will
it will
give
give
rise to
rise
a
to a
different
different
codon.
codon.
So it
So
is
it is
here
here
UUG.
UUG.
So
So
instead of
instead of
coding
coding
here
here
phenylalanine,
phenylalanine,
it
it
will be
will be
coding
coding
for
for
leucine.
leucine.
So
So
it is
it is
making a
making a
mutation,
mutation,
but
but
it
it
is
is
actually
actually
because
because
of
of
a
a
frame
frame
shift
shift
while
while
reading the
reading the
mRNA
mRNA
codon.
codon.
Okay, so
Okay,
these
so
are
these are
different
different
frames.
frames.
So
So
three
three
forward,
forward,
three
three
reverse. So
reverse. So
six
six
frames
frames
while
while
doing the
doing the
translation
translation
of the
of the
ribosome.
ribosome.
So
So
there is
there
a
is a
change in
change
the
in the
frame
frame
by
by
which it
which
is
it is
reading the
reading the
amino
the
nucleotide
nucleotide
sequence.
sequence.
That's why
That's why
this is
this is
frame
frame
shift.
shift.
Then
Then
again
again
you
you
can
can
actually do
actually
a
do a
base
base
pair
pair
insertion.
insertion.
So
So
here
here
you are
you are
inserting
inserting
a
an
nucleotide
nucleotide
here.
here. So
Then
then
it is
it is
actually
actually
coding
coding
here
here
for a
for a
stop
stop
codon. Just
codon. Just
after
after
methionine,
methionine,
it
it
will
will
stop the
stop the
protein
protein
synthesis.
synthesis.
Then you
Then
can
you can
also
also
add
add
three
three
codons
codons
through
through
which you
which you
want
want
to
to
actually
actually
insert
insert
one of
one
the
of the
amino
amino
acids
acid
in
in
between
between
that
that
sequence.
sequence.
That
That
can
can be
also be
also be
done.
done.
So
So
in
in
all of
all of
this,
this,
there is
there
a
is a
shift
shift
in the
in the
frame
frame
of the
of the
translation.
translation.
That's why
That's
these
why
are
these are
known as
known as
the
the
frame
frame
shift
shift
mutations.
mutations.
So
So
all
all
these
these
are the
are the
different
different
methods
methods
of
of means
different
different
types of
types of
mutation.
mutation.
Now we
Now
are
we are
coming
coming
to
to
a
a
method
method
which is
which is
very
very
important for
important for
rational
rational
protein
protein
design.
design.
For
For
example, you
example, you
know
know
now
now the
protein
protein
structure,
structure,
and you
and you
know
know
that
that
this is
this
a
is a
binding
binding
pocket
pocket
which we
which we
want to
want to
mutate
mutate
a
a
certain
certain
amino
amino
acid
acid
which we
which we
want to
want to
mutate.
mutate.
So
So
suppose
suppose
there is
there
a
is a
hydrophilic
hydrophilic
amino
amino
acid,
acid,
and
and
if you
if you
mutate it
mutate
to
it
a
to a
hydrophobic
hydrophobic
amino
amino
acid,
acid,
it
it
will
will
interact with
interact with
its
its
ligand
ligand
more
more
strongly.
strongly.
So
So
then
then
you
you
want to
want to
specifically
specifically
mutate
mutate
that
that
hydrophilic
hydrophilic
amino
amino
acid,
acid,
not
not
other
other
amino
amino
acids
acids
in
in
this
this
rational
rational
protein
protein
design.
design.
So
So
then
then
what you
what you
will
will
do,
do,
you
you
need
need
certain
certain a
technique
technique
through
through
which
which
you
you
can
can
introduce
introduce that specific mutation in that particular amino acid that we are interested upon, not certain random mutagenesis. So for that, people have came up with this technique of site-directed mutagenesis. So what people do is that this is your means first you need to clone your means you need to have your gene. For example, you are working on a protein. For example, we work on cadherines. So we have e cadherin. So we had to first clone my e cadherin into a expression vector. Okay. So first, after your cloning is successful, then you have a plasmid which have your e cadherin and your expression vector is carrying out this is carrying this e cadherin gene in it.
Now
Now
you
you
want to
want to
mutate
mutate
certain
certain
region
region in this
E cadherin
E-cadherin
gene.
gene.
So
So
suppose
suppose
you
you
consider
consider
this
this
plasmid
plasmid
as the
as the
plasmid
plasmid
of the
of the
E cadherin
E-cadherin
flowed
flowed
into
into
a,
a
specific
specific
vector,
vector,
expression
expression
vector.
vector.
So,
So,
but
but
in this
in
E
this
cadherin, you
E-cadherin, you
want to
want to
mutate a
mutate a
particular
particular
residue.
residue.
Okay.
Okay.
So
So,
for
for
that,
that,
what
what
you
you
do
do
is
is
that
that
you
you
design
design
two
two
primers.
primers.
Here
Here,
there is
there is
no
no
reverse
reverse
or
or
forward
forward
primer.
primer.
What
What
you
you
do
do
is
is
that
that
this only
only
this
this
amino
amino
acid you
acid you
want to
want to
mutate.
mutate.
So
So,
keeping
keeping
the
the
other
other
amino
amino
acid
acids
in
in
its
its
two
two
ends
ends,
constant,
constants,
means
means
not in
not
the
in the
amino
amino
acid,
acids,
in the
in the
codon
codon
level, in
level,
the
in the
nucleotide
nucleotide
level. You
level.
are
You are
making the
making the
primers.
primers.
So
So,
you
you
are
are
not
not
changing the
changing the
sequence
sequence
in the
in the
either
either
side
side
of
of
this
this
region
region
which you
which you
want to
want to
mutate.
mutate.
There can
There
be
can be
15
15
nucleotides
nucleotides in
in
this
this
side
side
and
and
in
15
15
to
to
18
18
nucleotides
nucleotides
in
in
this
this
side
side
which are
which are
exactly
exactly
matching
matching
with
with
the
the
or
or
which are
which are
complementary
complementary
to
to
the
the
nucleotides
nucleotides
which are
which are
present
present
in that
in that
particular
particular
strand.
strand.
Only
Only
the
the
mutation,
mutation,
only
only
the
the
only the
only the
region
region
or the
or the
amino
amino
acid
acid
which you
which you
want to
want to
mutate,
mutate,
that
that
amino
amino
acid
acid
codon,
codon,
means
means
the
that the
codon
codon
of that
of that
amino
amino
acid
acid
is
is
being
being
replaced
replaced
in
in
your
your
primer,
primer,
which is
which is
being
being which is being
marked
marked
by
by
a
a
cross
cross
here.
here.
Okay,
Okay.
so
So
then
then
what you
what you
will
will
do
do
is
is
that
that
you
you
will
will
carry
carry
out
out
this
this
PCR.
PCR.
You will
We will
carry
carry
out a
out a
PCR
PCR
with these
with
two
these two
primers
primers in
but
but
the
the
enzymes
enzymes
here
here
what we
what we
use
use
is
is
not the
not the
normal
normal
the
means
tag
the tag
polymerase.
polymerase.
Since
Since
it's a
it's a
mutation that we
mutation
are
that we are
designing, and
designing,
here
and
there is
here
a
there is a
linear
linear
amplification that
amplification that will be
happening,
happening,
since there
since
is
there
a
is a
amino
amino acid
there
since
is a
there is a
nucleotide
nucleotide
mismatch
mismatch
while the
while the
amplification.
amplification.
So here
So
we
here we
use
use
a
a
enzyme
enzyme
has a
which has a
higher
higher
fidelity
fidelity
or a
or a
higher
higher
processivity,
processivity which is
less
less
error prone.
error prone.
That is
That is
PFU
PFU
turbo we
turbo we
use
use
normally.
normally.
Okay,
Okay.
so this
So this
PFU
PFU
enzyme,
enzyme,
this is
this is
used
used
for this
for this
amplification.
amplification.
Then
Then
from that
from
you
that you
PCR
PCR
amplify
amplify
your
your pins
construct from
construct
this
from this
parental
parental
template,
template,
and you
and
will
you will
now
now
get
get
two
two
products.
products.
One
One
which will not carry out, which will
which will
not be
not be
having this
having this
mutation.
mutation,
That is
that
your
is your
parental
parental
template.
template.
Another
Another
is
is
the
the
mutated
mutated
gene or
gene
the
or the
mutated
mutated
plasmid.
plasmid.
Now you
Now you
don't
don't
want
want
to
to
select,
select,
you
you
don't
don't
want to
want to
have your
have your
parental
parental
plasmid
plasmid
in
in
that
that
mixture of
mixture of
the
the
parental
parental
as
as
well
well
as
as
mutated
mutated
plasmid.
plasmid.
So
So
for
for
that,
that,
people
people
came up
came
with
up
the
with the
idea
idea
that
that
there is
there is
a
a
particular
particular
enzyme
enzyme which
which
is
is
this
DPN1,
DPN1,
which
which
came
came
from a
from a
bacteria
bacteria
known as
known as
Diplococcus
Diplococcus
pneumonia,
pneumonia,
and
and
then
then
from
from
where
where
they
they
have
have
this
this
DPN1.
DPN1.
DPN1 has
DPN1
a
has a
particular
particular
feature
feature
is
is
that
that
if
if
your
your means
your
your
strands
strands
are
are
or
or
your
your
nucleotides
nucleotides
are
are
methylated
methylated
or
or
hemimethylated,
hemimethylated,
it can
it can
specifically
specifically
recognize
recognize
that
that
methylated
methylated and
hemimethylated
hemimethylated
tag
tag
in
in
a DNA,
DNA,
and it
and it
can
can
digest it.
digest it.
So
So
it's a
it's a
nature
nature
of
of means
in the
in the
bacterial,
bacterial means
when
when
this
this
replication
replication
happens,
happens,
as
as
well
well
as
as your
plasmids
plasmids
are
are
means
means
actually
actually
replicating,
replicating,
transcribing in
transcribing
the
in a
bacterial
bacterial
genome,
genome.
there
There
is
is
methylation
methylation
and
and the
different
different
demethylation,
demethylation, methylation
methylation
and the
hemimethylation
hemimethylation
happens
happens
in the
in the
bacterial
bacterial
genome.
genome.
So
So
the
the
once
once
this
since
this
this
template
template is was
present
present
in a
in a
bacterial
bacterial
genome,
genome,
the
the
template
template
will
will
carry
carry
this
this
methylation
methylation
marks,
marks,
whereas
whereas
the
the
product
product
that
that
you
you
actually
actually
amplified
amplified
from the
from the
template,
template,
they
they
will
will
not
not
carry
carry
out,
out, they
they will
will
not be
not be
having
having
any
any
methylation
methylation
in
in
their
their
nucleotide
nucleotide
sequence.
sequence.
Means
Means
the
the
mutated
mutated
product will
product will
not have
not have
any
any
methylation,
methylation,
whereas the
whereas the
parental
parental
will
will
have
have
a
a
methylation
methylations
in
in
it.
it.
So
So
that's
that's
why
why
after
after
linear
linear
amplification,
amplification,
you
if
can
you can
see a
see a
band
band if that's your means mutated means you will be seeing two bands. One is of your template and one is of your nicked product. Since there is this will continue till this and this will again continue till this, and there is a mismatch in this region, so there will be a nick created in this region, single stranded nick, which that will be repaired while we transform it into a bacterial strain. So what we do is, after you see amplification, you digest the plasmid with the DPN1. So the parental strand will be digested, whereas the mutated strands or the mutated gene, they will be intact. And you isolate and you take this plasmid and transform it into a E. coli strain, and then you actually get this means then you carry out this normal expressions in since it was in an expression vector already you will express and get your mutated protein. So this is the basic principle of site directed mutagenesis. So what are the requirement here? The requirements, the basic requirements here are the you need to have a clone DNA, and you need to have an expression system where after making these mutants you can be able to express your protein. So this was all about rational designing. Now, just quickly giving you a summary about the directed evolution process. So this directed evolution in the history also we discussed that Francis A. Arnold, he reported the directed evolution of subtilis in E to obtain an enzyme means subtilisin E to obtain an enzyme variant which was active in a highly unnatural denaturing environment, that is at high concentration of polar organic solvent dimethyl formamide. So he actually by through the directed evolution approach for the first time he worked on the subtilisin E to obtain an enzyme which has a 2500 fold, if I remember, more efficiency than the normal enzyme that was previously known. So how what are the different steps in a directed evolution technique? So first here in the rational designing we were doing site directed mutagenesis, but here since we are not starting with a mean structure model or we don't know the function, just like nature, what we want to introduce, we actually introduce random mutagenesis by different random mutagenesis processes and recombination processes. Then those genes are being the mutants are being inserted into the bacteria, we get the proteins, and we actually carry out different selections and screening criteria based on their binding or as well as their enzymatic activities. Then those mutants which are actually giving positive results or having higher enzymatic activities, as you see this is an iterative process that I was talking about. This will again be carried out, again the mutation will be done, again the screening will be done, and it will done to that point till the point where you are not getting the best best function of your protein. Then that mutant will be selected and then it will be applied in several in application based processes. So these are the different steps that you can go through as well while reading. Okay, so the different steps are the changed enzymes are means the first you need to provide the random mutation. The second step is the genes are inserted in the bacteria which use them as template and produce randomly mutated enzymes. So here I am taking the example of an enzyme to make you understand this process of directed evolution. Then the charged enzymes are tested. The changed enzymes are tested. The enzymes which are actually changed means they are mutated. They are tested. Those that are more efficient at catalyzing the desired chemical reaction. So here, since we are using an enzyme, we are looking into the enzyme activity. Those are selected, and new random mutations are introduced in the genes for the selected enzyme, and the cycle begins again. So this is an iterative process that actually goes on until you get a best function of your protein. Now, so there are this directed evolution has been done by different techniques. And now since there are different complex processes which are not easily done by directed evolution techniques, so people came up with the machine learning, using machine learning in directed evolution as well, machine learning methods in directed evolution as well as so For that, you need to understand the process in which machine learning is actually playing a very important role in carrying out this directed evolution and performing this protein engineering process. That process is known as epistasis. So what is epistasis? Epistasis is a phenomena in genetics in which the effect of a gene mutation is dependent on the presence or absence of mutations in one or more other genes, respectively, and and respectively termed as modifier genes. So here what it basically talks about is that the mutation of a particular gene, their function is actually regulated by the mutation of another set of gene, which is actually creating a genetic environment around that particular gene which is carrying out that mutation. Means the only mutation of a particular gene will not be sufficient to provide the function. It is also its genetic environment which is actually responsible for either for giving out the function. So the effect of the mutation is depend on the genetic background in which it appears. So you will be able to understand by this example. Suppose there is a normally people have black hair, sometimes brown hair as well. But let us consider normal wild type is they have people have black hair. Okay. Now that there is a mutation in that gene. Okay, and it is causing to certain means color change of their hair. Either it is yellow or red. Means depending upon different mutations in their gene. But if there is another mutation which will actually lead to baldness of that person, then that mutation will actually overcome the mutations of these two colored hairs, because there will be that mutations is actually leading to the baldness of the person. But there is a point mutation in the gene which is coding for the different color of the hair, but ultimately since it has another gene for the baldness, then it will actually somehow masking the effect of the mutations in the color of the gene which is responsible for the color of the hair. Okay, so that is a classic example. Here also you can see that this b is actually mediating or what kind of result it will actually take place. Okay, so this is the modifier. If you consider this b in terms of genetic level, it is known as the modifier gene, which actually modifies the function or the mutations which happens in another gene. Okay, so that is this is a quite a complex phenomena, since here only just mutating a single gene will not give us the function, because other genes are also responsible for creating this genetic background. And for this solving this kind of problem in directed evolution, people came up with different machine learning methods. Okay, so now Prince, I will discuss with you one of the application of this computational modeling. So that is known as the iterative protein redesign and optimization suite of programs, which is also known as the IPRO. So the IPRO redesigns proteins to increase or give specificity to native or novel substrates and cofactors. So this scheme or this suit was previously designed to actually see whether how to provide insights about the increased usage of certain cofactors in a protein structure. As you know that these cofactors are very important for the enzymatic activities of many enzymes. Okay, so the more easily like sir talked about this cytochrome P450, which he worked in IIT Roorkee. So where he was talking about the different this has a porphyrin ring which binds to iron. So if there are other cofactors which it can carry forward, and that can actually increase the activity of the oxygenase activity of the cytochrome, then it can actually, this is an example of protein engineering. So for solving those things, so this IPRO program suite is designed. Okay, This is done by repeatedly randomly perturbing the structure of the proteins around specific design positions. So you know the specific design positions where your cofactors or your binding pocket is there, where your cofactors will bind. So you are just perturbing the structure of the proteins in those positions, and you are seeing whether there is any effect in the binding pocket which is being created in the protein, okay, which can acquire different cofactors or different ligands, novel substrates or ligands. Identifying the lowest energy combination of rotamers, because your energy should be minimized always, and determining whether the new design has a lower binding energy than prior ones, whether this newer design is much means efficient or lower design, lower energy than the previously available structure. So what are the different functions of EPRO? So it can lead to the altering protein binding affinity and specificity. Okay, so there is a pore designer which is being created, which is a derived program from EPRO. So there you know that there are different bacterial pores, means the pores of the different channels are present. So those can be actually modulated with the help of this Pro Designer, which is a derived program of EPRO. Okay, so what are the different functions? So it is altering, it can be used for altering protein binding and affinity and specificity. It can be also used for enhancing enzymatic activity. It can also use for grafting a binding pocket that I was talking about into an existing protein scaffold, predicting an antibody's tertiary structure, and also assessing the structure and binding energetics for a specific mutant. So these are all the functions that this EPRO can carry out. So this is all about that I wanted to discuss with you, means a summarized version of what sir has talked about in week 11. So yes, so now I had some practice questions for you. So before that, if you have any questions, you can ask me, or we can directly go to solving the practice questions.
Sir,
Yes, yes,
can you
Sharon.
can you please repeat once more why the anti-parallel strands of sorry, why an anti-parallel beta sheets are more stable than the parallel beta sheets?
Yes, so in the anti-parallel beta sheets, if you see, if I go back mm, to that slide. Hmm. So here, what you see here is that these hydrogen bonds, which are being connected to form the ultimate beta sheet by two different beta strand here. So in the anti-parallel one, you see these hydrogen bonds are parallel to one another. They are coplanar with one another. Means if a bond is like this, means is parallel to one another and they are collinear with one another, then there is less restraint on that structure. Means they can withstand more forces than a parallel structure where you see that also this means there can be some means steric clashes in this parallel beta sheet. And another major reason is these hydrogen bonds, they are at an angle, they are not as strong like the anti-parallel beta sheets where they are collinear with one another, they are parallel to one another. So the strength is much more in case of anti-parallel beta strands, whereas in parallel beta strands, since they are in an angle and there can be also a chance of steric clash, so that's why these parallel beta strands are less stable than as we compare with the anti-parallel beta strands. Okay.
Okay, so there is a chance of the steric repulsion in case of the parallel beta sheets, right?
Yes, there is a chance, and also these hydrogen bonds that you see here is that the hydrogen bonds are not collinear with one another. There is a strain, there is an angle between these hydrogen bonds.
Angle
So
actually.
yes, so if they are in an angle, then their strength is not that much strong as we compare with the anti-parallel beta sheets, where you see all these hydrogen bonds are parallel with one another, they are collinear with one another.
Okay,
Okay,
so
so they are holding
the
the
two
two
beta
beta
strands
strands
much
much
stronger
stronger
than
than the
a parallel
parallel
beta
beta
strand.
strands.
If
If
we
we
have
have the same number of hydrogen bonds,
same
same number of residues.
okay,
Okay,
so that
so
is one of
that
the
is one of the
reason why
reason why antiparallel beta strands are more
stable.
stable.
Okay,
Okay,
Okay, sir. Thank you.
okay.
okay.
Any
Any
more
more
questions?
questions?
So I would like to add a little bit.
Yeah.
Sharanya, you know amyloid structure. Amyloid structure. There is mostly antiparallel beta sheets. They actually like work as a dentate structure. Like the side chains are they are protruding in that way. They will like fit into each other like two antiparallel beta sheets. They will fit into each other like a teeth-like structure. So that's why it is very difficult to degrade that structure. That's why it is very difficult to degrade amyloid structure, which is formed in case of Alzheimer's, Parkinson's.
Yeah.
Yeah,
Yeah.
beta amyloid structure, right?
Yes, yes. There also you will get the mostly antiparallel beta sheets.
Yeah.
Yeah.
Okay, okay.
So I
So
think
I think
Paramita
Paramita
works
works
with
with
beta
beta
amyloids,
amyloids,
right?
right?
So
So
that's
that's
why
why
she
she
can.
can give
Yes,
us.
yes, yes, yes.
Yeah.
Okay.
Okay.
So
So
yes,
yes,
it's
it's a nice discussion.
so
So
yes.
yes,
Any
any
other
other
question?
question?
Not from my side.
Okay.
Okay, okay.
So
So
now
now
let us
let us
just then
just then
quickly
quickly
go
go
to
to
some
some
practice
practice
questions
questions that can
that
that will
will
help
help
you
you to
to
actually
actually
solve
solve
the
the
questions
questions in your
your
exams, as well as the
assignment
assignment
already
already you have
submitted.
submitted.
So
So
these are
these
the
are the
questions.
questions.
You
You
can
can
just
just
click
click
a
a
picture,
picture,
or you
or
can
you
just
can just
solve
solve
now
now
and
and
keep your
keep your
answers with
answers
you.
with you.
When I
When I will be
discussing the
discussing the
answers, you
answers, you
can
can just
check
check
how
how
many
many
answers
answers
you
you
have
have
attended
attended
correctly.
correctly.
So
So
yeah,
yeah, how many
that's
questions you have attended correctly?
so
So
yeah.
yeah,
I will
I
give
will
you
give you
2 minutes.
2 minutes.
Just
Just
go
go
through
through them
and
and
try to
try to
solve
solve
them.
them.
These are the
These are the
questions
questions from the
previous
previous years, right?
previous years
Previous year's
assignment.
assignment. So I had
Got some.
got some access to it.
So
So from there, from
those I
those, I
actually
actually
make
make these
practice
practice
questions
questions
to
to
you
you,
so
so
that
that
you
you
will
will
have
have
an
an
idea
idea
about
about
this
this
year's
year's
assignment as
assignment
well
as
as
well as
previous
previous year's assignment,
which can
which can
actually
actually
help you
help you in your exams
if there
if
is
there is
similar
similar questions may be asked
in your
in your
exam.
exam.
So
So
for
for
that
that,
you can
you can go through these practice questions in
each of
each of the
slides
slides that I have
shared
shared with you in your
Google
Google
Drive.
Drive, in the Google Drive.
Okay.
Okay.
Shomodeep, I will request
Yes.
one thing. If it
Yeah.
is possible, if you can like suggest any book or like links where we can
Mm.
practice more structural biology MCQ questions, so that I can improve my knowledge as well as understanding. If you get to know, please share. Okay,
Okay.
Okay.
any
Sure.
book
Sure.
or links.
Yeah.
Yeah,
So
so
there
there
are
are
very
very
good
good
books.
books.
So
So
I will share
I will share with you
the
in the
exercise
exercise
of
of
each
each
of
of
that.
that,
They
means
are
they are
book of
book of
structural
structural
biology.
biology basically.
In the
In the
exercise
exercise you may get some of the questions, not
MCQ,
MCQ. I will say that
there
there
can
can be
be
some
questions
questions
which
which
will
will
have
have
direct
direct answers.
but
But
yeah,
yeah, I
I
can
can
share with
do
you.
I
I
can
can look into
some of
some of
the
the
practice
practice
problems
problems
based
based on what is
there
there
in
in
this
this structural biology course,
and
and
surely I
surely I
can
can
discuss with
discuss with
you
you,
I
or I
can
can
share with
share
you
with you,
or
or
what I
what
can
I
do
can
is
do
that
is that
I
I will
take
take
one
one
revision
revision
class
class
on
on
22nd.
22nd.
Okay,
Okay,
so
so means
21st
21st
I will
I will
take
take
this
the
week
week
12
12
discussion.
discussion.
We
We will
will
do the week 12 discussion,
and
and on 22nd I will be taking a revision class that will help you in your exam, solving your exam. So so I think in that class also I can give you more problems, because there I will not be explaining you the means concepts. Rather, we can focus more on the solving different questions in that class. So that I will try.
okay.
Okay.
Sure, sure. Because just like if reading something, it is not sufficient to know
Yeah,
Yeah,
if
yeah,
yeah,
whether your
yeah.
knowledge is like if your understanding is clear or not. So,
Yes, yes, yes.
okay,
Okay,
okay.
okay. I will definitely. That's a good suggestion. I will definitely
Do
do
that
that
and
and
share with
share
you.
with you.
Okay.
Okay.
Yes,
Yes,
Sharon.
Sharon. No,
You
you
want to
want
add?
to add something?
Yes, nothing but it's very helpful if we get one MCQ link. It will for the NPTEL exam. It will be very much helpful for NPTEL exam.
Yes,
Yes,
yes.
yes. I just like Paramita said that
I
I
will
will
try
try
to
to
compile
compile many as many MCQ
questions
questions
as
as I
I
can
can
find
find
regarding
the
means the
topics
topics
that I
that I
had
had
covered,
covered means
the
the
contents
contents
that
that
sir has
sir has
provided with
provided
you,
with you,
and
and
I
I
will
will either
share with
share
you,
with you
and
and
also I
also
will
I will
discuss
discuss
the
the
questions
questions
in the
in the
1 hour
1 hour
class
class
that I
that I
will
will be
taking in
taking
the
in the
revision
revision
class.
class 22nd.
Okay,
Okay,
so
so I think
that
that
will
will
be
be
good
good
for
for
you.
you.
Okay,
Okay.
okay,
Okay,
fine.
fine.
So
So
already I
already I
think
think
one of
one of
you
you
has
have
already
already
answered.
answered.
Amardeep
Amardeep
has
has
already
already
given
given
the
the
answers
answers
in
in
the
the
chat
chat
box,
box,
I
I
think.
think.
Uh,
Um,
yeah.
yeah.
Yes.
Yes.
So
So what he has given,
I will
I
check
will
your
check your
answers
answers
as
as
we
we
complete
complete
all
all
these
these means
practice
practice
questions. So
questions. So
there are
there are
four,
more
five
five
questions. So
questions.
you
So you
can
can
again
again
solve
solve
them
them
and
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 and again
write it
write it
here,
here,
or
or
you
you
can
can
check it
check it
by
by
yourself
yourself
once I
once I
discuss the
discuss the
answers.
answers.
Okay.
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 Okay,
So
so
I
I
think you
think
have
you have
solved the
solved the
questions
questions
or
or
clicked
clicked
a
a
photo.
photo.
You
You
will
will obviously get this
the
in the
slides
slides
in
in
the
the Google Drive,
which
which
I
I
will
will
share
share
with
with
you.
you.
So
So
yeah,
yeah,
let's
let's
discuss the
discuss the
answer,
answer,
and
and
I
I
will
will
also
also
check
check
Amardeep's
Amardeep's
answer
answer
what
what
he has
he has
given.
given.
So
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 the
the yes, the
first
first
question was
question was
what does
what
the
does the
Ramachandran
Ramachandran
plot
plot
show?
show?
So
So
as
as
we
we
discussed,
discussed,
it's
it's
the
the
frequency of
frequency of
phi
phi
and
and
psi
psi
values
values
of
of
the
the
residues
residues, amino acid
So
residues.
the
So the
answer
answer
is
is
D.
D.
Which
Which
factors
factors
contribute
contribute
to the
to the
stability of
stability of
protein
protein
structure?
structure?
So
So,
yes,
yes,
all
all
these
these
factors:
factors,
hydrophobicity,
hydrophobicity,
electrostatic
electrostatic
interactions,
interactions,
and
and disulfide bridges. So, this is
quite
quite
simple. We
simple.
have
We have been
discussing
discussing
about
about
this
this
from the
from a
lot of
lot of
discussion
discussion
sessions
sessions
that we
that we
had
had
previously.
previously.
So the
So, the
answer is
answer is
D,
D,
all of
all
the
of the
above.
above.
Then which
Then
of
which
the
of the
following is
following is
not a
not a
component of
component of
PCR
PCR
reaction?
reaction?
If
If
you
you
remember,
remember,
I
means I forgot to
talk
talk
about
about
PCR
PCR
in
in
this
this
class,
class, but
yeah,
yeah,
means
means
what are
what are
the
the
means
means
different
different
components
components?
if
If
you
you
remember
remember
the
the
PCR
PCR
when
when
we
we
talked
talked
about
about
PCR?
PCR,
So
so
flanking
flanking
primers
primers
you
you
require
require
for a
for a
normal
normal
PCR
PCR
reaction.
reaction,
Then
then
you
you
will
will
require
require
DNTP.
DNTP. You will
You will
definitely
require a
require a
template
template
DNA.
DNA.
What you
What you
don't
don't
want
want
in your
in your
PCR
PCR
reaction
reaction
is
is
the
the
DNS,
DNS,
since
since
it will
it will
digest
digest
your
your
DNA
DNA
and
and it will
not
not be
the
the
PCR will
PCR will
not
not
happen.
happen.
So the
So the answer is again D.
so
So the answer
the
here is D.
the
The
rational
rational
design of
design of
new
new
protein
protein
molecules
molecules
to
to
design
design
novel
novel
activity,
activity,
behavior,
behavior,
or
or
purpose,
purpose,
and to
and to
advance
advance
basic
basic
understanding of
understanding of
protein
protein
function
function
is
is
called as.
called as.
So
So
it's
it's
not
not
protein
protein
instability,
instability,
it's
it's
not
not
protein
protein
unfolding,
unfolding,
and it's
and it's
not
not
protein
protein
cleavage.
cleavage.
So
So
you are
you are
designing
designing
the
the
proteins.
proteins.
Remember the
Remember the
different
different
techniques
techniques
of
of
protein
protein
and
and
variant
variant
designs. I
designs.
have
I have
said that
said that
this
this
is
is
also
also a
part of
part of
rational
rational
designing.
designing.
So
So
the
the
answer
answer will
will
be
be A.
A.
Okay.
Okay.
And what
And
is
what is
de
de
novo
novo
design in
design in
protein
protein
engineering? De
engineering? De
novo
novo
means
means
starting
starting
from
from
a
a
new
new
sequence
sequence
based on
based on
prior
prior
sequence.
sequence.
Okay,
Okay,
means
means
designing a
designing a
new
new
sequence
sequence
based on
based on
prior
prior
sequence.
sequence.
Designing
Designing
of
of
protein
protein with
with few
few
mutated
mutated
residues
residues
with the
with the
original
original
sequence
sequence.
is
This is
not de
not de
novo.
novo.
Okay.
Okay,
So
so
de
de
novo
novo
means
means
from
from
you are
you are
designing
designing
a
a
new
new
sequence
sequence
based
based
on a
on a
certain
certain
prior
prior
sequence,
sequence,
but
but
you
you
are
are
designing
designing
from
from
scratch.
scratch.
Okay.
Okay,
So
so
that is
that is the
the
answer will be
here
here
B.
B.
Okay.
Okay,
Which of
which
the
of the
following
following
is the
is the
protein
protein
structure
structure
prediction?
prediction?
Uh,
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 so
so, uh,
someone
someone
has
has
given
given
about
about
the
the
answer
answer
as
as
A.
A.
Okay.
Okay,
With
with
few
few
mutated
mutated
residues
residues
in
in
the
the
original
original
sequence.
sequence.
No,
No,
I
I
think
think
the
the
answer
answer
is
is
here
here
B.
B.
Okay.
Okay, so designing a new
Sequence
sequence
based
based on prior sequence.
which of
Which
the
of the
following
following
is the
is the
protein
protein
structure
structure
prediction
prediction
method?
method?
So
So,
so
the
the
structure
structure
prediction
prediction method
method
is
is
okay. So
okay. So
here
here
I
I
have
have
not
not
given you
given
the
you the
answer.
answer.
So
So this
I
I
need to
need to
change.
change.
Which of
Which
the
of the
following
following
is
is
the
the
protein
protein
structure
structure
prediction
prediction
method?
method? So
It's a
it's a
protein
protein
structure
structure
prediction
prediction
method.
method.
So
So the here
here
is
the
the
answer
answer
here
here will be homology modeling,
right?
right?
Means
Means
the
the
sequence
sequence
alignment, the
alignment, the
phylogenetic
phylogenetic
evolution,
evolution, protein purification. These are not giving you a prediction method.
the
So the
homology
homology modeling is a prediction
so
method.
the
So the
answer
answer is
is
B. Yes,
Amardeep
Amardeep has
correctly
correctly
said that.
said that.
So
So
I
ITaser
structure
structure prediction
is
is
based
based
on
on means
I
ITaser structure prediction is based on what? It's
based
based
on
on
the
the
multiple
multiple
threading
threading
approach.
approach.
Remember
Remember the
the threading
threading we first
the
for
first
selecting the template there will be threading.
okay. So
Okay, so
it's a
it's a
multiple
multiple
threading
threading
approach
approach
that is
that is
followed.
followed.
So the
So the
answer
answer
is
is
C.
C.
What is
What is
silent
silent
mutation?
mutation?
Change
Change
in
in
nucleotide
nucleotide
sequence with
sequence with
no
no
consequence for
consequence
an
for an
amino
amino
acid
acid
in
in
protein
protein
sequence.
sequence.
I
I
just
just
explained
explained you
while
while
going
going
through
through
the
the
class.
class. So the answer here is
B,
B,
right?
right?
Change
Change
of
of
one
one
nucleotide
nucleotide
in the
in the gene sequence of an
enzyme
enzyme
is
is
known as
known as
substitution.
substitution.
So
So again, the
answer
answer here is
B.
B.
Okay.
Okay,
So
so and the
last
last
question
question
is
is
that
that
epistasis
epistasis
is
is
a
a
phenomenon
phenomena
observed
observed
in
in
what?
what?
So
So it's in the genetic
phenomena
phenomena that is happening.
the
So the
answer
answer
is
here is
B.
B.
So
So
these
these
are
are
all
all
the
the
practice
practice
questions.
questions.
Now
Now
quickly
quickly discussing discussing with
those
you
are
the
questions
questions
of
of
your
your assignment
week
means week
11.
11.
Okay.
Okay,
So which
so
of
which
the
of the
following is
following is
responsible for
responsible for
specifying the
specifying the
3D
3D
shape of
shape
a
of a
protein?
protein?
So
So
the
the
3D
3D
shape of
shape
a
of a
protein
protein
is
is
actually
actually
determined
determined
by
by
the
the means
amino
amino acid
sequences
sequences
which
which
is
is
present
present like
Finson has
Finson has
already
already
said
said
that
that how the protein
fold
will
it's
fold,
written
it's
in its
written in its
primary amino
primary
acid
amino acid
sequence.
sequence.
The answer
The
is
answer
D.
is D.
Which of the
Which of
following
the following
factor is
factor
not
is not
responsible for the
responsible for the
denaturation of
denaturation of
proteins?
proteins?
So all
So
these
all
heat,
these heat,
pH, and
pH, and or the
solvent are
solvent, they are more important for
how the
how the
protein
protein will be
present in a
present in a
folded form
folded
or
form or
unfolded form.
unfolded form. But
Charge is
charge
not
is not
responsible for
responsible for the
the
denaturation of your protein.
so
So
the answer
so the answer is B.
so
So in
around the
around the
Ramachandran plot,
Ramachandran
the
plot,
values
the
of
values
phi
of
and
phi
psi.
and psi.
So
So if you remember,
I
I
have
have
also
also
written
written so
C
C
alpha
alpha
and C
and C
alpha
alpha
n.
n.
So
So
answer is
answer is
B
B
here.
here.
Which of
Which
the
of the
following is
following
the
is the
basic
basic
requirement of
requirement of
PCR
PCR
reaction?
reaction?
Two
Two
oligonucleotide
oligonucleotide
primers,
primers,
DNA
DNA
segment,
segment,
DNA
DNA
polymerase,
polymerase,
and
and means
DNA
DNA
dependent
dependent DNA
polymerase is
polymerase
required.
is required.
Heat stable
Heat
DNA
stable DNA
polymerase is
polymerase is
required, like
required,
the tag
like the tag
polymerase.
polymerase.
And so the
And
answer
so the
is D.
answer here is D.
What are the
What are the
requirements
requirements
for
for the
directed
directed
mutagenesis?
mutagenesis?
So
So
remember, I have
remember, I had
written
also
these two
written these two
points. So
points.
site
So
directed
site directed
mutagenesis,
mutagenesis,
you
you
need
need
a
a
cloned
cloned
DNA
DNA
previously,
previously,
and also
and
you
also
need
you need an expression system
where you
where
want
you
to
want to
express
express
your
your
mutants
mutants
and
and
see
see
whether
whether they are
then
where
you
then
will
you will
screen them.
screen them.
Okay.
Okay,
The answer
the
here
answer
is
here
both
is
A
both
and
A
B.
and B, so the answer is C.
What are the
What
two
are the
general
two general
strategies for
strategies
protein
for protein
engineering?
engineering?
National
National
protein
protein
design and
design and
directed
directed
evolution.
evolution.
Answer is
Answer
A.
is A.
Which of the
Which of the
following is
following
not
is
true
not true
about ab
about ab
initio
initio
predictions? The
predictions?
ab
The
initio
ab
prediction
initio
method
prediction method
attempts to
attempts to
produce
produce
all
all
atom
atom
protein
protein
models
models
based on
based on
sequence
sequence
information
information
alone with
alone
some
with
aid
some
of
aid of
protein
known protein
structure.
structures.
So
So
ab
ab
initio
initio,
it's
it's
not
not
based on
based on
only a
only a
sequence
sequence
information,
information,
it's
it's
more
more
based on
based
some
on some
energy
energy
field
fields that you need to consider, and also the conformational space. So this statement is wrong. So the seventh answer is C. In Rosetta, the segments with assigned secondary structure are subsequently assembled into. So in Rosetta, you do three-dimensional protein modeling. So the answer is here B. So three. What is the iterative protein redesign and optimization method? So it's if you remember the definition there. So it redesigns protein to decrease or give specificity to native substrates or cofactors. So the answer is A. And the last question is means change of one nucleotide A to C. It's like it will give it will give substitution. The process is substitution here. The answer is B here.
Okay,
Okay,
so
so
thank
thank
you,
you,
and
and
I
I
have
have
noted
noted
down
down
your
your
insights.
insights.
So
So
yes,
yes,
I
I
will
will
prepare
prepare
one
one
questionnaire
questionnaire
maybe
maybe for a revision
class,
class, and
and
also
also
I
I
will
will share
share with you
the
the
means
pins
questions.
questions.
Okay,
Okay,
in
in
the
the
revision
revision
class,
class,
or
or I can share you
before
before
also,
also.
just
Just
look
look
into the
into the
Google
Google
Drive. I
Drive.
will
I will
share with
share
you.
with you.
Okay.
Okay.
Uh,
Um,
Eighth question, I
so
have doubt.
yeah, yeah.
yeah, Amardeep, say tell.
Eighth, eighth question, I have doubt. From
Which
Which
one?
one?
In
last
Means
the
in the
practice
practice question
or in
or
the
in the
last?
last question?
Oh, in
Oh,
the
in the
assignment.
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 Okay.
Okay.
So
So
Uh,
what
what
doubt you
doubt
have?
you have?
Ninth question, what is iterative protein design?
Yeah,
Yeah,
yeah.
yeah.
So
So
it
it
tells you
tells you
that
that
the
the
designs
redesigns
of
of
protein
redesigns proteins to
decrease
decrease
or
or
give
give
specificity
specificity
to
to
native
native
substrates
substrates
or
or
uh,
uh,
okay.
okay,
So
so
it's
it's
wrongly.
wrongly. It's
I
I have marked it wrongly. It will be
it will be
C,
right.
right? You are telling you that the answer will be C.
Yes, sir.
Yes,
Yes, yes. So I think I have wrongly marked it.
the
The
answer
answer
given to
given to
you
you
is
is
A or
A or
C.
C.
The answer is C.
C,
C,
right?
right?
Okay,
Okay,
I
okay.
have
I have
wrongly
wrongly
marked it.
marked it then.
Sorry,
Sorry, sorry.
that's
That's
my
my
mistake.
mistake.
So the
So the
answer
answer is redesign of proteins. Yes, it's not decrease. It should be always increase or give specificity to the native or novel substrates, not decrease. So then what's the why we are doing this optimization method? Why we are carrying out?
thank you,
Thank you,
Amardeep,
Amardeep,
for
for
pointing
pointing
this.
this.
I will
I will
correct
correct
it
it
and
and then give you.
but I
But I
think the
think the
answer
answer is
given to
given
you
to you
already.
already.
So the
So the
answer
answer
is
is C.
so
So
that
that
is
is mistake from my
mistake.
side.
So
So
the
the answer is C here.
okay.
Okay.
So
So
yeah.
yeah.
Okay.
Okay. So I
If you
if
people
people don't have much
doubts,
doubts,
then
then we can end the session here and we can meet
again
again
the
the
next
next
Friday.
Friday.
Okay.
Okay.
So
So
Yes.
so
bye.
bye.
Okay.
Okay.
Bye.
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 Okay.
Okay.